And excellent. So, and please tag us on X or on Blue Sky, hashtag HCA 2025 GM. Be a 3-day meeting or 3.5-day meeting starting today. And all right, I mentioned this already. I should mention also I'm Shyam Prabhakar, Genome Institute of Singapore, and various committees in the Human Cell Atlas. My colleague Shin at GIS, also a key part of the HCA. Big thanks to our platinum sponsor, 10x Genomics, for supporting this meeting. Also our gold sponsors, Atrandy, AWS, Helmsley, Illumina, Roche, and many more sponsors. We have silver sponsors, 12 of them, and our bronze sponsors as well. Thank you to all of them, and over to you. Oh yes, and continuous support from CZI and Klarman Family Foundation, and welcome for very, very generously supporting the core operations of the Human Cell Atlas, without which none of this would be possible.
Yeah, thanks, Shyam, and my name is Shin from Genome Institute of Singapore, and also want to welcome all of you to Singapore, and we're looking forward to the next few days. Again, just a few housekeeping rules, and just keep you guys up to date so that we are all safe and sound. So if there are any concerns, please do email Christine or myself. COVID-19 and other illness, hopefully everyone's okay, but we are still mindful, so please be aware of potential symptoms and emergency and question contacts. And I'm not changing slides, but changing on its own. Wear your name badges at all times. Ensure, obviously, you have, this will give you a pass to various places, and also there are QR codes that you can scan for contact. Just a key code of conduct is inclusivity, accessibility, respect, and please do not harass. And again, we are all wonderful people in this room, so I believe this won't be a big of a concern. So we really look forward to really constructive discussions in the next few days. And I just want to give you a quick overview of our layout. It's a little bit chaotic, but here on the top left is the auditorium. That red cross is where your registration was. I just want to highlight that we have an editor from Nature Methods here, and Madhura is willing to meet with all of you during all break and lunch sessions, and she'll be right at the registration desk. So if you want to talk to the editor, please do so during the break sessions. Wi-Fi, very important. Please take note, pictures if you need to, but I think it's no password needed, so I think it's no issue at all. Let me see. Believe it or not, there are charging devices, there are charging sockets around you, so please use it. Please do not leave your belongings in the room, and you can actually drink, and food and beverage are permitted in our rooms, including the auditorium. But please be respectful. Oh, we're missing a picture of Shyam, but you know his face. So just want a big thanks to all the, there you go. The superstar always comes at last. And 25 HCA General Program Committee, thanks so much for all your efforts to putting this meeting together. And I won't name all the names here, but you get to meet them throughout the next few days. So thanks again. And again, the meeting themes for the next few days is really to build, continue to build roadmaps and in building atlases. And you'll hear right after us from Aviv and Sarah on the opening remarks about the current status and the direction of building atlas. And key theme here is spatial omics, and you'll hear that a lot. And hopefully there'll be more participation and contributions from this aspect. Human diversity is key importance for us in the Human Cell Atlas, including equity and cohort engagement, and how to really dive into disease applications along with healthy atlases. So I think these are really exciting times for single cell and spatial omics, and we really look forward to your active participation. So yeah, today we have a full day, and we again look forward to your contributions. Right after us, there'll be welcoming remarks, and there'll be various, again, coffee breaks, Meet the Editor, along with key sessions on spatial. And we'll have a very key note from Sonia and technology showcase, followed by breakout sessions, which is really the heart of Human Atlas Consortium meetings, where these breakout sessions will really get all your inputs into an action item. So with that, we'll close with a reception and some poster session. So once again, I just really hope that you guys will spend the next few days to connect with people and socialize, and hopefully there'll be new collaborations coming out. So with that, I'll invite Shyam back to introduce the next session. Thank you.
Thank you, Jay. If we haven't said it already, this is the first Human Cell Atlas General Meeting in Singapore. We're very thrilled to have it here. Our two opening speakers for the opening session are none other than Sarah Teichmann, who's here, and Aviv Regev, who I think is online, to give you the opening view. Thank you.
Hey everyone! Hopefully you can hear and see me. If not, just let me know. I'm welcoming everyone to the Human Cell Atlas General Meeting, and I want to say first of all that unfortunately this is the first time that I'm actually missing an in-person general meeting. I had to stay kind of at the last minute in California this week, but I look forward to several really great days of virtual participation, and will do my best, given the time zone differences, to at least enjoy all the mornings. So let me start by anchoring us in our mission. It is to create comprehensive reference maps of all human cells, which are the fundamental human units of life, as a basis for both understanding health and diagnosing, monitoring, and treating disease. And I think that it's interesting to think on achieving this mission, that we really rely on a recipe that HCA has honed over the years, or kind of our not so secret sauce. And it's a very simple recipe. It only has 6 ingredients. So unprecedented scale, global experts working with rigor and quality. And by using all of these things, we deliver groundbreaking science, we develop lab and computational methods, and we really bring in the end tangible biomedical impact. And I'm going to take a moment to dig through each of these. So first, for scale, the HCA members have generated the largest single cell atlas as a map of human cells. And it's enabled research that was impossible with smaller data sets. And that includes, everything from the scientific discoveries that were made to what we're seeing now in the training of foundation models on HCA data. We also work globally and openly, and I would say more so than any predecessor consortium that has existed in our fields. And that means that we have more than 4,000 members across 104 countries right now. And this means that we have both the world's leading experts and expansive geographical representation. And also, we do our work with rigor and with quality. It means that we're defining the cutting edge, and we also stay at the technological cutting edge. We standardize data formats and metadata, and we ensure rigorous, expert-defined atlas standards. As a result of all of this, people can really trust the atlas, and they can drive real impact for biology using the Human Cell Atlas. And that's really the kind of impact that we're seeing more and more now, that we're really achieving. And so the Human Cell Atlas led to the discovery of numerous cell types and states. It also characterized the cells and the states that it discovered. It helped define differentiation paths in processes that we really had no access to before, like human development. It uncovered how tissue is organized in molecular and in cellular terms. And it defined kind of a scientific playbook of how to do so. And that's all reflected not just in the 455 papers that are badged HCA and the 78,000 papers that cite them, but in the many, many pieces of science that has been done in those citing papers and in many, many others that are following this HCA playbook and are building on it. Now, at the same time, HCA member labs also developed the experimental and computational methods that underpin our entire field. And that includes numerous lab techniques for single cell and spatial genomics, RNA and chromatin and DNA and mitochondria and more and more and more. And also, all the way to computational methods that now expand to AI-based foundation models and agents. And that means that this ensures that this ever-growing atlas is organized and queryable and can be even filled by AI for the pieces that we can't finish measuring. And then finally, this yields every day tangible biomedical impacts. Scientists now routinely map genetic variants of disease risk to the cells in which they act. And they do that through the Human Cell Atlas. It wasn't actually doable before. Scientists use the Human Cell Atlas to nominate therapeutic targets and to understand how they might act and to assess if they can be targeted safely in patients. Scientists use the Healthy Atlas as a reference when they aim to engineer new cell therapies or new organoids or to run screens to revert disease cell states to healthy states. And they use the Human Cell Atlas when they nominate biomarkers that they would use in clinical trials in patients and in clinical diagnostics. And they do this every single day. Okay, so clearly the Human Cell Atlas is already transforming our understanding of biology and disease, and in this it's helping us revolutionize science and medicine. So I would say that's kind of a nice recipe, right? Just with 6 ingredients. So now let's see what our members have actually been up to. Now you might recall that we have 18 biological networks and each of them is focused on a different organ or system. And I'm really thrilled to update that 6 new version 1 atlases are now assembled and approved, and I'm going to take you through each one of them very briefly. So first up, the retina or the eye atlas. So Rui Chen and Ayelet Segre are the network coordinators. Fabian Theis was the computational integration PI, and the team is led by Jin Li, Lun Wang, Ignacio Ibarra, and many others. Let's start with the fact that this atlas identified 120 cell types in the retina and the eye, the vast majority of them different types of neurons. This is 50% more than what was previously known. And this integration was done across cell and nucleus profiles. It was actually the way in which we tested the approach to do so. It was done across tissues also, and it's multimodal, and that includes both RNA and chromatin and other features. And in this way, it went all the way to find gene regulatory networks that are associated with these many neuron cell types that were discovered in the atlas. All the data are in the portal, and the integration paper is actually available as a preprint on bioRxiv. Okay, next up, the liver. The coordinators for the BioNetwork are Alan Mullen and Gary Bader, and Gary Bader is also the computational integration PI. And a really amazing team is in the lead: Daniel Teichmann, Delaran Puyabahar, Gao Ran, Hongru Hu, Jordan Portman, Niranjan Srigaonkar, Rachel Edgar, Shruti Kare, Tallulah Andrews, Yongjin Choi, and others. I deliberately am reading all of these names because, of course, without everyone's work, this would not have happened. And again, 18 new cell types have been discovered in this process. Single cells and nuclei and the annotation and building were done by engaging a really wide community that brought a lot of expert knowledge and trust in the ultimate outcome. Now let's move on to the organoid atlas. And so the coordinators for the network are Christoph Bock and Barbara Treutlein. And then an exciting team participated with Fabian Theis from the computational side, along with Jisong He, Xuan Hu, Leonard Halle, Leander Doni, Jonas Fleck, and Gray Camp. They've integrated millions of cell profiles from neuroectoderm and endoderm organoids. And because these are lab models, it was really crucial to include many diverse models of human organ physiology that people try to do in the lab and to integrate them together. And that was actually very challenging because, of course, these experiments are not all identical to each other. But once the atlas was built, it allowed comparing the models to patient tissues, and that lets us know which model is good to model which purpose, which aspect of the human tissue, but also how to improve the models, essentially benchmarking new organoid protocols through this atlas. Again, the atlas is available on the portal, and it's described in 2 papers that are already published as part of our Nature package. Okay, next up is the gut. Matthias Zillbauer, Jose Ordovas Montañez, Michael Snyder, and Ramnik Xavier are the coordinators. Sarah, together with Fabian Theis, are the computational integration PIs, and Kyle Kimmler, Chris Lance, and many others are on the team. Millions of cells coming from almost 30 data sets and almost 300 individuals are the basis of this integrated atlas, which comes across a huge organ of the gastrointestinal tract. And this required organizing almost a comparably huge community and also developing new methods that will be able to streamline the data and the metadata QC. And then finally, no, not finally, one but last, another major organ is the kidney. Here the coordinators are Anna Greca, Matthias Kretzler, and Mina Clatworthy. Sarah, together with John Marioni, are the computational integration PI, and the team include Konstantinos Stasinos, Penghe, Rajasri Menon, and Aishwarya Subramanian. Similarly to the gut, almost 20 data sets from 200 donors. A community effort was needed in order to support the re-annotation, and as a result, this now established a gold standard benchmark for integration. And one key piece was actually linking the developmental trajectory of cell types and states. And now, last but not least, is the Human Development Atlas. The coordinators are Alain Chadotal, Diane Taylor, and Maz Haniffa, and in the integration, Maz, Fabian, and Sarah on the computational integration, with a great team including Anthony Rose, Kemal Inechik, Simone Webb, Chuan Shu, Emily Stevenson, and others. This is by far the most comprehensive developmental atlas of human development. And honestly, there weren't really atlases of human development before. One of the key innovations of the team was annotating the data in a way that allowed linking cell profiles from pre-implantation to data from later stages of human development. And also, the team now connected these data to the Decipher database on human developmental disorders. Okay, so let me pause here for a moment so that we can all appreciate this series of 6 amazing atlases. And I want to ask you to join me in applauding the coordinators, the computational integration PIs, and all of the team members and community contributors that actually made these atlases possible. Fantastic. Now, through the work on atlas integration, a lot of exciting computational tools have emerged that will enable not just work on the remaining atlases, but also the work of the community as a whole on many additional problems. So for example, MetaManager streamlines the collection and harmonization of metadata, which you know is absolutely essential for atlas assembly. SC-Atlas TB scales this iterative process of atlas building and evaluation. The integration team also constructed computational benchmarks to know whether the atlas is good or not, and that are optimized for millions of cells in order to reduce the amount of time that it takes for this iterative decision-making. And they put in automated and manual QCs, ways to harmonize labels, and approaches to integrate the ultimate data. Now, we also need to serve all this data to the world. And I'm also absolutely thrilled to announce that AWS, Amazon Web Services, will now be hosting the Human Cell Atlas on the AWS Open Data Registry webpage. And that means that people can access these data in a way that is highly accessible and very effective for them. It is a huge deal for HCA and many, many scientists across the world. We're very grateful to AWS for supporting HCA and its science in this way. And on day 3, we will actually have a workshop with AWS on how to unlock insights with the data using AWS cloud solutions for advanced omics and generative AI. We're also seeing the community increasingly relying on the atlases, not just to make new biological discoveries, but to develop the next generation of computational models and tools for biology. And for example, for training this next generation of models like foundation models. So in this case, we're seeing the promise of pairing AI with our cell atlases to ensure that they're accessible and that they're queryable by many people across many different areas of expertise, and that they empower those people in order to make really meaningful discoveries, both basic research and translational ones. And what we're seeing coming already behind it in the AI world are autonomous AI agents. So autonomous agents are equipped with foundation models, but they can make decisions, they have memory, and they can take actions on their own without needing constant human supervision and guidance. So an agent can kind of understand its environment, and this environment can include computational tools. And then it can take actions using those tools, run analysis, look at the results, reason on those results, and continue on the analysis. And in the last year, since our last meeting, multiple such agents have been released. For example, HCA labs, multiple HCA labs, have put out AI agents that focus on spatial analysis, a major theme of our meeting. These are 2 of them, spatial agent on the left and SP agent on the right. A very recent set of agents, like this agent here called Biomni, that comes from Yura Leskovich's lab in Stanford, are general purpose agents for biomedical research. Now we are seeing it executing some analysis around single cell RNA-seq of a small atlas of the gland, of a salivary gland. And so in this particular case, the agent has learned how to do single cell analysis by using an action discovery agent that read tens of thousands of publications across 25 biomedical domains, including our domains, with all these great HCA papers. And in this way, it mined essential tools and databases and protocols. And then it has this generalist architecture that allowed it to integrate LLM-based reasoning with retrieval augmented planning and execute code. And then it dynamically composed and carried out these complex biomedical workflows like the one that I just showed you for single cell genomics. Okay. Many of these innovations have been published in the past year, especially in our package of over 50 papers with Nature Publishing Group, which was featured simultaneously on the covers of Nature, Nature Medicine, Nature Genetics, and Nature Methods. And I know there is a Nature Methods editor in the audience. We reported in this package on large-scale data sets, on advanced AI methods, and many insights from human development to the host response to COVID-19. So lots of exciting progress. Here is our ask of you. 1, contribute your data and metadata when the bionetwork coordinators reach out. This is essential for them to be included in the atlas. 2, participate in the annotation jamborees. We need your expertise. This is essential if we want to make the atlas of high quality. And 3, we need you to be focused on finishing the atlases. And so if we call on you for help, please help us. Please help the bionetwork coordinators. Please help the integration leads. Please help the team members so that we can finish the remaining atlases. And with this, I'm gonna stop sharing and turn to Sarah to take us through HCA's priorities for the next 5 years.
Thank you, Aviv. Can everybody hear me? Yeah, great. I also can't resist telling the people at the back, there's lots of space at the top in case you want to sit. You can go up the stairs. So Aviv has really beautifully summarized our amazing progress to date. Over the past year especially, there have been so many exciting new advancements and progress in our community, and that's a super massive credit to everybody here and the broader community across the world. So looking forward into the next 5 years, what are our goals? Where should we be heading? So you've already heard about the reference atlases, and this is really a super valuable and precious resource that we are providing for the community. These integrated data sets that can be used as references for foundation models, for annotation, label transfer, and so on. And so obviously the next phase is going to be integrating those data sets across the human body to kind of provide one integrated human cell atlas reference. And we have a session today about roadmaps for building atlases relating to that. Then we also, of course, in the next 5 years, want to get to that atlas framework where we have X, Y, and Z in 3 dimensions, and have that map of where the cells sit in the tissues, in the organs, and ultimately across the whole human body. So we want a spatially resolved 3D atlas. Not sure if this pointer works. And again, we have a session today about spatial. And obviously, we've had massive tech advancements. We've got increasing data generation in spatially resolved tissue sections. So this is really where we are all heading together towards spatial resolution. And then what we also need to be cognizant of is that we have a representative atlas that represents humans around the world from different groups, and also a clinically applied atlas, because a lot of our impact is, of course, in terms of how our work and our knowledge then helps understand and treat disease. And we have sessions tomorrow on diversity and equity, and then on Wednesday on disease, sort of for this purpose. Okay, so cross-tissue cell atlas. Where do we need to go? So we are obviously in the process of this integration effort that Aviv mentioned and highlighted kind of for the first tranche of bionetwork atlases. We're now moving forward so that over the next 6 to 12 months, we'll have all 18 bionetworks having a reference, which we can then integrate into a single human cell atlas reference for suspension, cell, and nuclear data sets. We want to expand these in several dimensions. So capturing that diversity, achieving more comprehensive anatomical coverage, so every nook and cranny of the human body, so to speak, multimodal, so that we have also, multiplex kind of protein, RNA, epigenome, et cetera, representation, and more density in the number of cells so that we're also capturing more rare cell states. So this is really where we need to go with expansion. I've mentioned cross-tissue integration, of course, for the whole human cell atlas, so that we can relate the cell identities across organs and systems, because obviously, there are shared cell states for immunity, vasculature, nervous system, musculoskeletal, et cetera. What's shared and what's tissue specific? Kind of, this is what we can learn and understand in a way that's never been possible before through this exercise. And, as we've discussed, serving the data for the community is really going to be part of our impact, or a crucial part kind of into the future. Is we who work on these data sets, we're obviously deeply, intimately familiar with the data, the cells, et cetera. But we want to also allow everybody across the world to access this data, and in an easy kind of interactive way, like with that animation that was just shown with the agent, basically, for analysis and visualization. If we go on to the, yeah, okay. So our second goal needs to be achieving this spatial resolution, because obviously we are operating in 3D, our tissues, our organs, even in 4D, right? So how do we get to this spatially resolved reference with sort of multimodal? So obviously all the suspension cell and nuclear data sets, that are creating these references are going to inform basically our interpretation of the spatial data. But we need to understand where the cells sit in X, in Y, and Z. And so moving forwards into the spatial era, we want to expand the human cell atlas to achieve a comprehensive coverage in 3D, develop the spatial technologies, and also the computational tools that go with that, and of course then the portals. And finally, achieving an atlas that's globally representative and also has clinical utility. So there are two components to that, really, to that point. One is implementation of the Genetic Diversity Task Force, and this is important work, and Shyam and many others have worked very hard on this. Sarah Tishkov and the whole team, and we'll learn more about this at this meeting, to basically help us and guide us forwards in achieving more representation of groups around the world. And then secondly, also, there's been a lot of work in the community and also a lot of desire and motivation to use the Human Cell Atlas as a reference for understanding the changes in disease and in therapy, actually. And so having a framework to apply the healthy data in disease, but also in preclinical drug safety, so toxicity and safety is an obvious application even of the healthy reference atlas, is understanding where could side effects occur in the healthy tissues for drugs. So there's a very obvious, very clear kind of application, practical application of the Human Cell Atlas for disease. So I just want to summarize the feedback that you gave from the spatial survey and the disease survey, and thank you everybody who participated in this. And this was really to give us a summary of where the community stands in terms of spatial data, and the feedback was that there are two tissue preservation methods. Sort of more than half of the people had generated already spatial data with various technologies. Often it's paired with single cell or single nuclear data, kind of, so you have both. There are different tissue sampling approaches used, different sizes and sources of tissue, so whether it's resection, material, biopsy, et cetera. And then 3 different resolutions, so whether it's kind of regions that are larger than a single cell resolution, subcellular resolution, et cetera. And of course, we have a session later today about this. And thank you to Holger, Heyn for the spatial survey and analysis. And for the disease survey, and thank you, Shyam and colleagues for this disease-focused survey. The summary is about the diseases of focus, the numbers of tissues banked, cohort sizes, and what are the diseases that are prioritized by the bionetworks. And this is important to think about, because the priorities might be different, obviously, for different tissues, different places in the world, different research groups, and so on. And so this has been really useful in terms of also getting a feeling for which diseases the community wants to prioritize. So this is a really strong foundation now for going forwards in the next 5 years for delivering a comprehensive Human Cell Atlas. And of course, our foundation and our principles are strong governance, strong partnerships, planning, and thank you to everybody who's been involved in this. The data ecosystem, our central office that I'll come back to, who's amazing, and we're so grateful to them. Our community engagement, and of course, the ongoing support from our visionary funders. And you see a summary of these funders for the Healthy Human Cell Atlas and also for many disease cell atlases on this slide. And it's really, you know, it takes a village. It's really a massive effort, basically, and a massive support from many of our funders. And I'm just going to leave this up for a couple of minutes so that you appreciate kind of the diversity of support that we've had, which has made this possible kind of over the past many, many years. Now, we also have strong partnerships, and GA4GH is one example of that, which has been key for us to implement and develop our standards, all the single-cell consortia, the tech companies that have allowed us to power our data generation, and a massive thank you to all of the funders and all of our partners. I just want to also make you aware that we now have an industry partnership program, and there may be members here, and there are more companies joining this. The aim is basically to help exploring the Atlas applications in drug development, diagnostics development, a forum for knowledge exchange about the technologies and data analysis methods and so on, and also to facilitate the connections basically between HCA and pharma, and as a way of kind of expanding our impact. So we launched that last year. It's been incredibly successful, and there's been a lot of interest already, and this is actually growing as we speak. We've had for a long time, of course, the commercial discount program, and this is open to all of the HCA members, and here's the list of companies, just so that everybody is aware who's here and who's online. Again, this is expanded also with additional companies over the past year. So just to remind you, of course, everyone knows that we are a community, we work together, and we have really key partnerships that I've listed already, and we've had some additional ones over the last year. You've heard about HCA, you've heard about AWS, basically for data serving interaction analysis and so on. We hope this will kind of make things even more easy and streamlined going forwards. You'll hear in this meeting about our UNESCO partnership, powering open science, equitable contributions and benefits. And we're delighted that Casimiro is here, and there'll be an announcement with John Randall, who went to Paris for this event earlier this year, just a couple of months ago, and we're incredibly excited and honored to be partnering with UNESCO in terms of sort of the heritage of the Human Cell Atlas. So this is a real milestone for us, and we can all be very proud about this together. I've already mentioned our pharma partnerships, basically in translating, helping translate the research, and also from a safety point of view. And finally, our public engagement program, and this is really where the Human Cell Atlas reaches out to the public, to the communities, school students, interested public, via, for instance, science and art programs. So this is a little image from Bauman Leahy, which was from our Wellcome-funded One Cell at a Time public engagement program, and it's a really touching, beautiful image of an artistic interpretation of human tissues and cells. And we're very excited in the UK that the Science Museum in London is engaging with us for a Human Cell Atlas sort of exhibition, which is actually will be in the entrance hall and will start next year. And so that was, and I want to say a big thank you to the HCA Executive Office for supporting the engagement with that process, and also to many of the students and postdocs here, actually, who've been talking to the team at the Science Museum. So I just want to remind everybody of our governance. You know, we mentioned how important this is. We have the organizing committee, of course, which is sort of the central hub, if you like, of our leadership, our coordination. We have the expert working groups for analysis, ethics, equity, standards and technology, and data ecosystem oversight that are sort of cross-cutting, basically, for the whole community. And then we have the 18 biological networks that are the biomedical experts for the individual organs and systems. And you've heard that it's really a collaboration between the biological networks and the analysis groups to make these reference atlases. So we work in partnership across the community, and that's the beauty of this community that I love so much, is that we're interdisciplinary, and that's how we teamwork makes the dream work. Basically, that's how we move ahead together. So the executive office are these amazing team, the amazing staff that help us with our coordination, manage partnerships, and projects like the Science Museum and engagement. And here you see them. Some of them are here. Aya for the archive, who has also helped with public engagement. Yoshi is here, Esther, Anya, who has made these amazing slides. Bridget is here. Anne is not here for the web. John Randall, of course, is here and with Lucia leads this team. Christine is here for meetings, of course, is essential for this meeting and all of our meetings. And Ellen and Sam, I haven't seen here, but are incredibly valuable for the project management. Ellen for the publication bundle, and all of you will know her from coordinating that mammoth effort, and Sam for our communications. This is an animation from Anya about how we've grown as a community, and this has just been obviously incredible that we are now almost 4,000 members in over 100 countries. So, you know, we are a very large and strong community. And I will finish there and say thank you and how excited I am about the next few days, and looking forward to interacting with everyone and the sessions ahead. Thank you. So I'm going to hand over to our new vice chairs, Chloe Villani and Haniffa, and they're going to give us a session on the roadmaps for building those reference atlases. So it is a huge pleasure to introduce and hand over to Chloe. And you know, I'm very proud that the organizing committee has endorsed their important, both Chloe and Muz's important roles as and with the title of vice chair. So I will hand over, and looking forward to your talk, Chloe.
Thank you, Sarah. Okay, it is so great to be here. It's my favorite annual meeting. I get to see friends from all around the world, make new friends yearly. I really look forward to catching up in the coming days and meeting a lot of you throughout the different sessions. And so I'm going to pick up on where Sarah and Aviv left and dive into more of the operational aspect of our roadmap for building atlases. We're in such an exciting time right now. We moved over the years from our grassroots initiative, generating data, establishing a work group, to now assembling our atlases, right? So it's a phase 2, and this is where we're going to focus a lot of this session. While at the same time, we're planning the atlas expansion. You're going to hear more about this in the coming sessions today. And so I want to take us back to the Venn diagram that Sarah walked us through, right? So this session will focus a lot on our cross-tissue healthy reference. And throughout this talk, I'm going to try to make the point of how this is our base foundation atlas that's really going to feed into our next phase. You're going to hear about it in next session with Holger Heyn about a spatially resolved atlas, as well as a representative and clinically applied atlas, while gathering all of this data to build foundation models. And I want to try to explain to you how this is all related. So right now, we're really excited to working towards completing our version 1, we call it the base atlas. And to try to make it really functional and useful, we're also entering the phase of annotation and annotation jamboree, which is going to bring us to our version 2, and we're going to talk a lot about this in this current session. And once we have a very strong cross-tissue healthy atlas, this is going to set the foundation to develop panels, annotate our spatially resolved atlas, and really scale this up, both in the context of health and disease, which is also going to help us achieve our goal of creating a representative atlas. And through all of these different phases, we are actually gathering the data that is so key in fueling the next generation of HCA foundation models. But I want to start by focusing on our base atlas. And so the ultimate goal here is to create a cross-tissue 1D atlas for our first phase, right? We want a unified annotated reference map of the entire human body. So how do we get there? So first, you already heard in the introductory remarks that individual bio-network are generating their version 1 atlas. And there were very strict criteria defined by our organizing committee that are listed here, that every bio-network had to meet in order to get approval. That included all atlas had to be robust to technical factors. You couldn't just take the first integration method and first set of parameters out there. You actually had to test several of them and be very transparent on how you ended up selecting which method you used. You had to incorporate all the landmark studies, and this was really driven by our biological network. Every has to put their data, their annotation on the annotation platform. We're going to talk about this today. And all the data, all the files, all the code needs to be released publicly. And we had the ambitious goal that by this year, mid-year, which is going to be next month, we would have half of the atlases done. And actually, we are achieving this goal. We already have 8 biological networks that got approval by our organizing committee. And this is a huge achievement on its own, as Aviv mentioned, and you all applauded for it. It was not only that the community for this bio-network delivered, it's also all the work behind the scene that went into this. We revamped our data ecosystem, we developed new standards for our different type of metadata. There's a huge amount of work behind the scene that helped propel these atlases. And this is a roadmap for the remaining atlases. So you can see by July, we will have reached our goal of 50% of atlas approved, and we're working to being done by next year. Quite ambitious goal. And you're seeing different ones labeled in blue. Why they're so special is because we're going to talk about them in this particular session. But this is really key to make these base atlas very useful. We really need to kind of work towards the annotation. So you may all be looking at the same data. We may have used very similar integration pipeline, but when we look at the annotation, very often we don't annotate the same way. It can be a challenge, and that's a challenge we're going to be tackling. So how do we get there? I'm going to give you some specific examples in this talk and going to hear throughout the session. All the annotation for the integrated atlas are being deposited on the cell annotation platform. And then the community is invited to give feedback on annotation, both to derive consensus, but also define the areas of disagreement. They're as important, because once you define the areas of disagreement, we can also focus on trying to figure out why do we disagree. Maybe we need different type of data modalities to resolve these disagreements. We're going to then work towards cross-tissue integrations for these cells that are shared across organ system, like immune, adipose tissue, vasculature, stromal cells, and so forth. And then that's going to help us harmonize our cellular identity for these shared cell subset. There's a bit of a roadmap here. And so you can imagine that as we work towards this harmonized consensus annotation here at the bottom, that feeds in right into our next phase of informing our spatially resolved atlas and our disease atlas, and we're going to get back to this slide towards the end of the talk. And so why am I spending so much time saying like annotation is a challenge? That's because historically annotation has been more of an opinion than an objective science. Very often people have annotated their cells manually. It's not super scalable. We sometimes differ in the definition of what we think is a unique cellular identity. Sometimes we use different molecular signatures and markers. We use different vocabulary. We even may not be as precise. There's some lumpers and some splitters in the clusters, and that tends to be a problem. So how can we work towards standardizing this? This was the foundation for starting the cell annotation platform, which helps compile, compare cell annotation provided by different domain experts to derive consensus. And we're going to go in more details now. And we're going to invite the entire human cell atlas community to work with us, with every biological network. You pick your favorite cells to derive some consensus on the annotation to create our unified reference map. And so some of you might have heard me talk about this. So what is the Cell Annotation Platform? It's a community-driven platform to create, explore, and share annotation. We provide infrastructure to accumulate and analyze annotation. We are also developing new automated annotation services, and we have feedback features to help derive consensus. And we're going to give you some examples. And so to try to standardize the annotation process so that we can compare and contrast, we've defined some strict metadata structure so that everybody annotates with the same language. So we have 12 annotation metadata. I feel this is extremely well documented. They're listed here. It covers your name, your cell label, the marker genes. If you use automated versus manual annotation, for example, we cover if there's any ontology terms or what's the closest term. And this provides you the necessary context to consistently and reproducibly construct atlases and work with annotation. But it also gives you the metadata that you can leverage to compare and contrast annotation and gather feedback. And that's really key because now the data is structured. It makes it easier to compare our notes. And a lot of bio networks are already submitting their data on CAP. I want to do, since we're in Singapore, I want to give a big shout out to the Asian Immune Diversity Atlas. Ken and Eliara work really hard to put their data on CAP and many others. So big thank you for all of you. And this is for the Genetic Diversity Bio Network for pushing this forward. I won't go in great details, but actually you don't need to know a bioinformatics tool to engage with the annotation. With CAP, you have a user-friendly interface. You can work in private space. You can do live compute to annotate. We offer interactive UI where you can explore molecular signatures, pathways. You can calculate new differential expression, all for free. We're going to be implementing some annotation transfer very soon. We can look at the relationship between cell types, different data sets. And ultimately, once we reach some consensus, you'll be able to download cell cards. And so I want to show you some examples in practice. Some people are using the Cell Annotation Platform while they're building these atlases, and others are using them once they've built them. Skin is a good example. The Skin Bio Network has yet to complete their atlases, but they're using CAP to gather feedback on annotation. This is an effort led by the Bio Network coordinators Fiona Watts, Maria Casper, Max Plikus, with very close collaboration with Muzlifah. They've now integrated over 500,000 cells from different healthy skin tissue across 24 studies that are listed here, and to create this beautiful embedding. And for every one of these 10 lineages, they did different subclustering solutions to define different cell subsets. For example, zooming on the fibroblasts here, they have identified over 23 different subsets, and then they upload their whole embedding. We're going to show you a video on CAP, and then even include the doublets in there so that the embedding doesn't change as you get information and feedback. And these are little blue circles. They keep all the cells in. So what does it look like? So they were very kind in setting up a video for us. Let's see if this works. Okay, so I'm showing you the embedding of all of the cells, and on the left you can see they're clicking on different cellular resolution. They're zooming in on the fibroblasts here, and once you zoom on the fibroblasts, you get all the different clusters they define for the fibroblasts, and they provide you on the left with different clustering resolutions for you to go and annotate with the cells. So you can pick your favorite clustering resolutions. And then they've also provided different marker genes to try to show you that there are groups of cells with distinct molecular signature. This is, of course, a lot in the transcriptional space, showing you that there are indeed distinct fibroblasts. And so what you can do is go and look at the data, go and see if you agree with the different clustering resolution they've defined for the fibroblasts, and play with the data yourself by picking your favorite clustering resolution and giving them feedback. That's one way of approaching it. Now you have 12 different metadata. That's how we can get everyone's feedback. We can go and vote on them. We have established these feedback features. I'm going to give you some example. The first one is very easy. There's a thumb up icon. You can actually agree. Agree with all of it. Agree these cells are same identity. I agree with your metadata. This is right. Thumbs up. I won't show you a demo for this, but there's a lot of demo available on our website. You can say, you know what, I agree that from a transcriptional standpoint, these cells have the same cellular identity. But I disagree on how you define them looking at the metadata. Maybe you say, I want you to really build a cluster. Maybe you disagree with the markers that people are using or the cell ontology term people are using. Well, you can give that feedback. For example, for every cells, you get the full list of metadata. You could click on Refine Metadata at the top, and then you can scroll through the metadata. You could remove synonyms. Of course, it's under your name. You're not affecting the master file. And then you can go through all of it, and when you're done, you can send your change, and your feedback will have been recorded. You can also disagree. You can say, actually, you're wrong. These cells don't have, either you can say these cells are over clusters, this is a unique cell identity, it should be merged, or you know what, you're missing some subsets in there. The cell should be split. You can also record that feedback. This is an example from the gut cell atlas. So what you would do, you would select a specific cell. Here we're picking up T cells, basically saying like you should have really subclustered your T cells are more than a T cell subset. So you can select your T cells. I'm showing you there's some general markers on the right. You can select them, for example. And then at the very bottom right, you can provide feedback. You can say, I disagree. You say here, I want you to split. And then you need to tell us by how many groups of cells you want to split them. You need to give a justification and a list of marker genes and save your feedback. And all that feedback will be recorded. I'll give you an example of the liver atlas. In Aviv stock and Astara stock, you saw that the liver was approved. They've actually, their atlas is now on CAP. They are reporting over 500,000 cells, describing over 48 cell types. This was done across 6 different research centers. And so this was led by Yanyi Huang and Gary Bader, involving over 20 scientists from different institutions to deliver this work. So they've put their data sets on CAP. You have a summary of their study. And at the bottom, you have the data set across all the different cell lineages. You have annotated from the cholangiocyte, the mesenchymal cells, the myeloid cells, for example, the lymphocytes, the endothelial cells, and the hepatocytes, to name a few. And so if you go back to the myeloid cells, you can say, I want to explore the data sets. I want to look at the embedding. You can click on the embedding. I'm going to look at the myeloid cells. And they want to showcase, for example, for this example, that they've discovered new monocyte subsets. It's called a BAC3 positive monocytes. They're zooming in here to show it to you. And they're going to try to convince you that this is right. So you can select them, right? There's currently no feedback, so they invite you to give any feedback. And so you can select these cells. And then it's going to show you all the metadata they have to convince you this is a unique cell subset, including the marker genes that includes BAC3. You can click on this to see the marker genes that define these cells. And if you click on BAC3, it's going to show you that BAC3 is uniquely expressed in these cells. Now you can decide if you agree or disagree by selecting these cells, by clicking on it. I'm selecting them. And then I can go and provide feedback. Obviously, I know it's a lot to remember. The point here is not for you to remember all these instructions, but to show you the different capabilities of the platform. We're offering different tutorials and hands-on guidance to help everybody use these platforms, also through the bio network. Once we've gathered all the feedback, you can actually see the feedback summary. This is an example from the gut cell atlas. This is just a mock example. You get all the list of feedback, whether there's disagreement and agreement. You can also say, I actually just not sure, unknown. This is a question mark, for example. And so obviously this is Evan, who's our lead scientist, who wants to give you a demo. And then you can also filter based on agreement or disagreement to see the summary as a list, but you can also look at the molecular data, and you can actually project on your molecular data the areas where you see agreement or disagreement, and it kind of help contextualize where we may find some good alignment. So this is just one example. And so at the end, you get the feedback summary for each cell type. You get the feedback summary on the entire embedding. All the authors and the annotations are transparent and public. To give feedback, you actually need to register. It's very easy, just an email for registering. And as much or as little information we want to provide, but it's public. Review of your cell annotation is also public and transparent. And as we do so, it does provide us a framework towards identifying where we have areas of agreement and areas of a disagreement. And where we have areas of disagreement is where we can start identifying areas of consensus, which is really key. It's also providing us a huge database of information about annotation. It's important to find the areas where we find a lot of synonyms. We might have been walking around calling the same cells 20 different ways, and knowing the 20 different ways is super helpful. With all this information, that will also help improve a lot of automated annotation service moving forward. And so once we get some areas of consensus, we'll be building a lot of these cell cards. I will summarize all of this key metadata information inspired from gene cards. And so you're going to get the summary from the annotation terms, the synonyms, the marker genes, for example, and where this information came from, among other things. And so how do we connect building this consensus annotation to our next phase, right? For example, building panels for spatially resolved atlases. Because it would be very easy for many different individual labs to build their own separate panels, marker panels for spatial. But this would be a huge disservice to our community because it's a huge amount of work to do data integration. And we're having it easy. We're just doing single cell RNA sequencing suspension. And a lot of it was done using very similar technology. With spatial, there's a need to align. So with all these approved atlases or annotation jamborees and the consensus card, we can also try to see through our cross-issue integration how we can harmonize annotation. And we can use this to design our marker panels for spatial analysis and help align all the biological network, including for sets of markers for cells that are shared across organ systems, such as immune, stromal, adipocytes, and vasculature. And once we have these sets of panels, it actually really helps us to also scale up and empower downstream analysis, both for spatial and disease-related atlases. And that's what I meant when I say working with the harmonized and consensus annotation across tissue really help us propel our next iteration of atlases, building on our base atlas for the spatially resolved one and the clinically applied atlases. So there's a lot of documentation to work around with CAP, both in terms of the metadata, but also in terms of the wranglers. There's teams that are here to help support. A big shout out to Evan Bittersens, who's our lead scientist, and Mary Fute, who's our lead wrangler. They're available to help and assist you. We also want to hear about you and the feature requests. You need to help us help you by telling us what you really need as you use the CAP browser. A special thanks to Lyra and Yanyi Huang for their close partnerships, to Lichia and Ellen from the HCA Executive Office and helping us bridge with the data ecosystem. Felix Fischer, really talented PhD students from Fabian Theis's group, who's been helping us developing new analytical features on CAP. The whole CAP team, we have front-end, back-end software engineers, solution architects. There's a huge team behind the scene that has been working this for several years. And these are the CAP PIs. This was initially started by Peter Karchenko with Aviv, and John Marioni, Fabian and Niels Gettingsburg, who's here in the audience, and myself have taken the lead since our departure for industry. So with that, we'll reserve questions for the end. I'm really pleased to have a series of exciting speakers. They're going to highlight different aspects of different bio network and introduce each of them one at a time. We're going to start with Gary Reynolds. Gary Reynolds is a rheumatologist, physician, scientist. He's based out of Mass General Hospital. He's also the co-coordinator with me of the Immune Bio Network. I'm really pleased to have him here with us today.
Thank you very much. Thank you, Chloe. So I'm going to give an update of the Immune Cell Atlas version 1 on behalf of the Immune Bio Network. This has been a collaboration with the genetic diversity team headed by Shyam and the integration team headed by Malta. It's been an enormous amount of work by many of the people in the room today. It's a real pleasure to be able to see them face to face after so many Zoom meetings together. The Immune Bio Network now has more than 600 members over more than 40 countries. I'm going to talk about what we've been doing to date and what we plan to do in the future. And if you're not a member and this sounds like something you'd like to get involved with, then please do reach out to us on this email address. So the objective of the Immune Bio Network is to map the human immune system across tissues, across ethnicities, and across the lifespan of the human. This is an enormous undertaking, and so at the inception of the BioNetwork, we sought community discussion of how we should approach this. And so the theme is split into 4 areas to investigate blood, primary and secondary lymphoid organs, barrier tissues, and then finally all immune cells. And we've started in this atlas at blood, and that's partly practical and partly biological. Blood is a window into human health. It's the basis of one of the commonest diagnostic tests, but also there's abundant data that we can use as a proof of principle for generating the data. So the underlying hypothesis of this and the other atlases is that there are a finite number of biologically important immune cell states that will be conserved across health and disease and different diseases. So the constructed PBMC atlas now has more than 9 million cells. There's more than 2,600 donors, and it includes predominantly 2 conditions, so health and COVID-19. And the rationale for COVID-19 was, again, biology and practical. We understand the biology of COVID-19. There's been a lot of studies into it, and so we can assess whether or not we are recapitulating biology in the completed atlas for whether it recapitulates the biology we know should be there. But also, there are a lot of COVID-19 data sets, so that was why. These are the data sets we've included. Now we have remapped all of the data, and I'll explain a little about why we've chosen to take that approach. And we've also constructed all of the tier 1 metadata. The completed object has an almost 50-50 split of healthy and COVID-19, and we have a broad geographic representation, which we hope to expand further in the V2 Atlas. I've mentioned some of the ambitions for the Immune BioNetwork, and if you have data that you'd like to contribute, then also please do reach out. It's been something of a challenge to bring this data together and integrate it and develop a satisfactory atlas. The challenges we faced have been challenges that I think many people will have faced around scalability. So cell numbers, as cell numbers increase, it makes it more challenging computationally and time-wise. We've been optimizing each level of the atlas construct, and there are obviously many things that you can optimize, and so that's taken time. We have been reaching out to authors to gather metadata. Some of the published data lack some important information like sequencing batch. And as I say, we've gone back to the raw data because some of the published data was missing important lineage-defining genes like CLEC9A. And I think we discussed that a published object would not be acceptable without these. So this is an overview of the iterations we've been doing to assemble the atlas. As I say, we started from raw data. We've been optimizing how we approach quality control. We spent a lot of time discussing how we perform feature selection, so to remove batch effect, but also to make sure we're not losing disease biology. We've tried different integration tools. I think like many of the immune BioNetworks, we've settled on comparing Harmony and SCVI because of scalability issues with others. And as I say, we've been iterating all of the different parameters. For evaluation for each iteration, this is an overview of the approach we've been taking. We've been combining qualitative assessment. So this top left gives an example of one of the integrations that has these islands that are composed from a single data set or donor, which is obviously unacceptable. Because blood is so well studied, we know what we should be looking for, and so in the bottom left, we can see here that we would hope in the plasma cells we see a separation by heavy chain, and we're not seeing that, so we continue iterating. And alongside this, we've been using quantitative assessments. So I think, like many people, we've been using the SCIB package, but also developing novel metrics. So this is a metric that's based on looking for co-localization of well-localized genes based on Moran's eye. We have made a poster about how we've been iterating through this, presented by Sergio and Wamia, poster 37 today, so please do seek them out and have a chat about it. We have eventually settled on Harmony when comparing to SCVI, and these are the embeddings we've generated to date. Anna Maria is presenting a poster today about using the atlas to explore COVID-19. She's posted 9 today, so please do also go speak to Anna Maria. This is an example, the monocyte and DC2 embedding. This gives an example of the marker genes that we've chosen to define each lineage. And we've also been assessing our integrations by the degree of mixing. So we'd expect good mixing by chemistry, good mixing by data sets, and we have managed to achieve that, I think. As Sarah mentioned, we have been working together to try to improve how we iterate through different parameters. Michaela has built a toolbox for this, and she's going to be poster 68 today. Just say the data is available, or going to be available, on the HCA data repository, cell by gene, and the HCA data portal. So, as Chloe introduced, we've now been uploading the data onto the cell annotation platform, and I'd be really keen, we would all be keen to get your feedback on the annotations we've generated to date. So this is the celltype.info. This is the cell annotation platform homepage, and this is will bring you to the landing page for our V1 atlas. Each lineage will be put here. So this is the monocyte and DC2 object with the annotation we've ascribed to date with the relative proportions. You can explore the data set, and this gives you a summary of the information. This is the metadata, including important things like the batch that we've used. You can, if you wish, download the data in H5AD format and explore it however you like, but also in CAP, you can press molecular data and it will bring up the embedding for you to play with yourself. So this is the embedding with the annotation. This is the annotation on the side, so you can go into one of the populations. So as you can see, there's no feedback yet. And then if you click into Explore Metadata, this is some more information about the annotation and the marker genes we've used as evidence. You can show these genes on a heat map. So you can see the complement genes well expressed there. And then if you click on the dots, it will show you how they're expressed in the embedding. So then you can, as Chloe discussed, you can give us feedback on whether you think that's a reasonable annotation. So, as I say, for providing feedback, you can download the data and explore however you wish, or you can explore it in CAP itself. So the timeline, our next steps are to upload the other lineages. We're still coming to an agreement about the annotation. From August to September, those in the Immune Bio Network will start receiving emails inviting them to contribute to providing feedback. We'll be launching a Jamboree, which is going to be a combination of virtual and face-to-face, initially virtual to provide feedback on CAP. And then towards the end of the year, we will be comparing the results and trying to resolve any conflicts and come to an agreement about the annotation of the atlas. So as I say, if you'd like to participate, please do get in touch. As I say, we have posters about all the work we've been doing by the people who've been working on this project, so please do seek them out. So again, thank you to everybody in the Immune Bio Network for all of our hard work. Thank you very much.
It'd be great to have a handheld webcam. Okay, it's my pleasure to now introduce Ed Lein, who's a senior investigator at the Allen Institute in Seattle. Ed wears multiple hats with the Human Cell Atlas. He's a member of our organizing committee. He's also a member of the Data Ecosystem Oversight Group that Shyam and myself chair. He's also a co-coordinator of the Nervous System Bio Network. I'm really pleased to have Ed today to share with us some updates on their roadmap. So you have something?
Okay, can you hear me okay? Okay, I think we're go. All right, it's really a pleasure, and I'm grateful for the chance to come and visit Singapore for this meeting and see all of you again. I'd like to give a very brief update on creating and annotating the brain cell atlases in human and other species for this meeting. So first of all, I should say that most of what I'm going to talk about is actually done under the auspices of the NIH Brain Initiative Cell Census Project. This is a really well-organized consortium by the National Institutes of Health to try to bring together groups to really tackle this problem of mapping the brain. The brain is much more complicated than most other organs. For a human brain, it's quite inaccessible for study. And so actually a cross-species multimodal approach is really essential to tackle brain organization. This has been through two phases. One was called BICCN, or the Brain Initiative Cell Census Network, that was mostly focused on mouse brain and trying to scale methods to work in human brain. And now we're in the next phase called the Brain Initiative Cell Atlas Network, or BICAN, which is predominantly focused on human brain. I just want to step back for a second and say, like these cell atlases are really important for neuroscience and have the potential to be really transformative for the system as a whole. And the goal of BICAN is to create this foundational reference that serves all parts of the community. So not just the single cell atlas part, but really the cell, the circuit, the developmental components, neuroimaging components, other major efforts to understand the wiring of brain cell types, or create genetic tools for genetic access or potentially therapies as well. And of course, this interfaces extremely nicely with the Human Cell Atlas and other consortia that are trying to do this across the entire body. For the most part, we've really been trying to follow some of the footprint of the genome projects. And what I mean by that is that the transcriptional approach or the genomic approach has been extremely effective at being able to classify cells. And we want to create a resource that has the same sort of reference properties that the genome has for the field as well. And that means moving from these single cell studies to really formal versions of these references that have formal nomenclatures. We have versioning associated with them. We have tools to access the data. We have tools to map against it and transfer labels onto your own data. It's really important for adoption for people to be able to actually use it. And then the knowledge aggregation, you sort of saw the idea of a cell type card there, but if you go to gene cards, there's an enormous amount of information that continues to be layered onto that reference. So that's the idea with these cell references. For brain, just a sort of extremely brief recap, the BICCN effort started in one part of the brain, in the mouse and in the monkey and in the human, and it really showed that you could use a transcriptionally based approach to map, to define the types of cells, their hierarchical relationships, layer onto that their spatial organization, different properties of the cells, and map across species. And this is a really important sort of a first foray into this to show that this would be meaningful and could be scaled to the entire brain. Just 2 years later, we had already achieved the map of the entire mouse brain and also a very good first draft of the human brain as well as part of this first effort. So this is really, I think, a testament to the technologies and their scalability that you can move very, very rapidly once you have the infrastructure set up to do this. The other thing I want to point out from this is that you really need both the single cell and the spatial for this to understand. And I don't see these as serial things anymore. You do them together, and you use that to help to annotate the process as much as possible. The other is that the brain really is extraordinarily complex. On the mouse side, over 5,000 clusters could be defined, and we now have an understanding of the typology and their spatial distribution across the brain as well. We're not quite there in the human. So now we're in the next phase called the Brain Initiative Cell Atlas Network. And this is now, as I mentioned, predominantly focused on human, but it has a series of different projects. The one that I'll predominantly focus on today is the one that we lead at the Allen Institute. And our project is really trying to get a map of the entire brain. So it's not a population study, it is a small number of individuals where we spend our resources to try to map the entire topography of the brain. I'll describe this in more detail, but there are other really important projects as part of this that go much deeper in different parts of the brain on epigenomic profiling, on looking at individual variation, and going into development, both in human and non-human primate. Compared to the first phase, this next stage of mapping is much more ambitious and much more complex. We're attempting to get comprehensive coverage across the large brains in human, macaque, and marmoset and map that onto the mouse that we already have. So this comparative strategy is baked into the whole approach. We're trying to do this with a multimodal approach as well. So not just the single cell multi-omics, but also spatial transcriptomics, I'll talk about a little bit, and other phenotypic characterization where we can. And this is actually where having the multiple species helps you, because we don't get access to tissue for that kind of characterization in human for very many parts of the brain, but we can do it in the non-human primates and use a homology mapping to infer properties to human. And then finally, to try to map this into a coordinate framework that's useful for the functional imaging community, to do this from the beginning, which required really going upstream in the process of how we prepare brains and how we document those and how we can map those into an MRI-based coordinate framework. So the three core goals, though, and I think this will be pretty common to any of the organ systems, are to get a brain-wide consensus cell classification. And in this case, this also means across species. We complement that with spatial maps of tissue microorganization with the spatial transcriptomics types of methods. And then these are put into common coordinate frameworks, which are the gross anatomy of the brain, that have a parallel architecture across species as well. And so we're actually doing this on MRI-based templates for human, macaque, and marmoset, and the mouse for that matter, where we've now also defined consensus anatomical structural ontologies that map across species. So at the end of the day, we're going to be able to map from human across model organisms that are common across the field and in biomedical research. So the Bicam Consortium decided to focus on one part of the brain to start. This is really important because the brain itself, as a whole, is extremely complicated. We have to take a sort of a targetable part of this to get the system set up. And so we decided to focus on the basal ganglia. This is a subcortical set of structures which is involved in motor control, cognitive affective control, and affected in a whole wide range of brain disorders as well. So this provided a really clinically relevant target for us to do, but also a more or less contiguous set of structures that we could use for profiling. The core elements of this have been a multi-omic analysis with, principally this is 10x multi-omics, so RNA-seq plus ATAC-seq, which we do in parallel across species, where we map across the topography of those structures in the 3D organization and integrate these data to map them together and really get this homology-based classification. Of course, in individual species, there are species specializations, there are levels which are difficult to align, but down to a certain level, it's really possible to do this alignment. We marry that with spatial transcriptomics based on marker panels derived from that classification, and have been doing this in parallel across the 3 species as well. And then we couple that also with this phenotypic characterization of cells, because many of these cells are actually not well known, or at least the properties of them have not well been described in primates or humans for sure. One point I wanted to make is that spatial methods are really difficult to apply to these larger brains, and this is true for other larger organs as well. And so, whereas a mouse fits in the commercial platforms and a marmoset just barely fits in, macaque doesn't, and human really doesn't. And so you end up having to do these sort of tiling strategies to span a particular brain region. And this is possible to do, and relevant for where we'll go in the future. So, for example, in a marmoset is small enough, we can simply sample across the entire organ. We don't actually need volumetric coverage to get a good sampling of that, but you do need regular coverage across the different structures. Macaque you can do with this tiling approach, but you don't need too many tiles. In fact, this is possible to scale to whole brains, so we're tackling that. But humans are different beasts. It's so much larger. Individual parts of these already took 9 tiles. We had to break it up into 9 pieces, section them all independently, and then sample them across Z. It's really not the way to go for whole brains, so we need new strategies and new technologies for that. So we have these data now, and now we've begun the process of trying to annotate and turn this into a really formal reference. And so now we have 4 species data for the omics. We can align these. We actually use the spatial information to help to understand whether this classification makes sense. Spatial organization and cross-species homology are powerful indications that these are important. We map it to the literature. And then on the other side, we have created the whole data structure and software tools to be able to move through a standardized set of pipelines, through new standards for taxonomy schema, using tools for annotation like CAP, as I'll come to in a moment, getting these into cell ontology, putting all this information into a brain knowledge platform so we can reveal all these with a fully featured set of resources for the community. ultimately, this ends up with this consensus taxonomy. And just one point I wanted to make here, if you work in one species, you might have one nomenclature, but if you work in multiple species, suddenly the perspective changes a bit, especially with molecular definitions, because markers often are not conserved. And so you don't want to use a marker that's not actually relevant for the species. If you have that information, you can put it into the definition in the first place and define a nomenclature system. At some level, for machine learning types of things, it just matters if it's machine readable, but people actually care about these things. They want a meaningful name for their type of cell. Okay, so we have begun this community annotation process as well, and I just really wanted to highlight the contributions of the CAP group for this. We've put this tool to use in our first Jamboree of this sort. This Jamboree was the first really going beyond the Brain Initiative effort. We used this with these references to try to discuss markers and clusters and splits and all the things that Chloe's mentioned. This was actually quite successful. We had lots of feedback coming from this, and I think we can use this not only sort of in person for this, but on an ongoing basis. These were of all the kinds of things you could imagine. People wanted to use it in different ways. They wanted to discuss particular splits. They wanted to discuss differences in nomenclature for these things. And so it really helps to illustrate the challenges and places where we can improve the taxonomy. I was asked to give some tips for the use of it and what actually came out of this, so much more informal, but I think that orchestrating these jamborees and using the community annotation is a work in progress, and it takes some serious coordination around this. I think what we learned from this is we should do a little bit more work up front to give some tutorials on how to use the platform, extra information for this annotation as we got people in place that they were already geared to go. I think one of the extra challenges is that we have actually all this supporting information. We've got the spatial organization, we've got the phenotypic information. How do you get that so that people can see all of it as they're evaluating whether this was a good molecular classification? But anyway, again, I want to make the point that I think these things are not done and you're finished with the job. It's going to be iterative over time, and CAP provides a nice way of accumulating that information over time. I wanted to mention just a couple of things to close here. The first is that we're coming to our own publication package on the basal ganglia. And this is the first one that I'm aware of where we've got a whole series of papers that all are focusing on this one part of the brain, and everyone agreed to map to the consensus basal ganglia cell classification. So even in the past, we've had 8 papers and they have 8 different cell definitions. So we can start to work towards using the same reference across studies. Maybe we can convince the rest of the community to do that as well. So that was just one part of the brain. And so in the next 2 + 1 years, we need to move to the rest of the brain, which is still a big challenge, but this is what we're supported by the Brain Initiative to do. And one thing I really want to highlight is that we're still in the state of a field, even among our own labs, where we create multiple versions of these things, and it becomes very challenging for people to know which one they should use. And that's very much the case here. We have a draft whole brain atlas. We have detailed parts of the atlas. We have disease studies that have gone really, really deep and have more information. And so we want to move towards a single version that really everyone can use and represents the current state of knowledge and an iteration process for this. But we've actually gotten into a different place where before we have somebody working on human and macaque and marmoset, and you start to put these things together over time. But now actually we have all the data together. And so we can now start with a cross-species unification to get a homology-based framework, and then you refine from there, and we can do these jamborees across different parts of the brain, and over time fill in the whole atlas. So with that, let me thank some people. This one project that I told you about is in itself already a very complicated consortium, an international consortium. It's a really incredible team effort to work on this project. This is part of a larger ecosystem of people and collaborators working with this. I'd like to thank our funding agencies at the Brain Initiative, NIA, and NIMH, and also collaborators that we didn't call out here, such as AWS, that have been really helpful. And then finally, I included a slide in particular to call out some of the other contributors and the CAP team who's helped to support this. Thank you very much.
Thank you, Ed, for a fantastic overview. I'll now welcome our last speaker, Professor Muzlifah, who's the newly appointed head of the Cellular Genomics program at the Wellcome Sanger Institute. She was also newly appointed with me vice chair of the organizing committee. She wears multiple hats, like all the speakers in this session for the Human Atlas Initiative. She is on the supervisory board of the Human Atlas Teaching, which is really key for us to be able to share data all around the world. She co-coordinates the Development Bio Network that you're going to hear about. She's also a member of both the Equity and the Data Ecosystem Oversight Group. We look forward to hearing your Atlas roadmap, Ms.
Thank you very much, Chloe, and it's a great pleasure to be here today. It's one of my favorite cities, very close to where I come from, Malaysia, so it's home away from home. So I'm going to talk about the developmental cell atlas integration. Oh, it's not actually projecting on. I can see my slides on here at least. So I'll start with the intro slide. So this is going to be about the developmental cell atlas integration, and I'm speaking on behalf of the entire bio-network. And the work has been primarily led by Anthony Rose, Kamal Inerchik, Simone Webb, Emily Stevenson, Chuang Zhu, with support from Martha Lucan, Fabian Theiss, Sarah Teichmann, and also myself, I suppose. So this is very much about the developmental integration, which is one of the 18 bio-networks within the Human Cell Atlas, as you know. It's still not displaying on the back, okay. Yeah, perfect. So one of the bio-networks, one of the 18 bio-networks. And so it's really exciting to be here to showcase what we've been doing. So the Developmental Biological Network is coordinated by Alan, myself, and Deanne Taylor. And as Gary and many of the other bio-networks have shown, please do contact us if you are interested in being part of the bio-network and contributing to the effort. So what we did actually about 2 or 3 years ago now was to assemble the integration effort and pull together studies that had good numbers of cells, captured a range of organs, and also complied with the Tier 1 metadata for the Human Cell Atlas. And this included 21 developmental organs from 188 donors, spanning 4 to 22 post-conception weeks with regards to the gestational stages, and included more than 3.6 million cells or nuclei. But you can see here immediately the challenges of this effort. Firstly, the fact that this is one of those cross-tissue integration. You have multiple organs here. You've also got a time component, and you have multiple platforms from which the data has been generated, and a huge number of cells, which creates the engineering problems of integration. And I'm just going to highlight some of the features of the data that we've integrated and showing you by gestational stage in the first instance. So you can see that each individual study captures different time points, and you might see that the organs have a preferential capture over a certain gestational stage than others, and that's primarily because this is the critical point for that organ development, or some organs don't exist after a certain stage, for example, the yolk sac, or some organs are only formed after a certain stage, for example, the thymus. So it spans the first up to the mid-second trimester. You can also now look at the number of cells and contribution by the organ with regards to how many samples and how many cells. So that's all of the features of the data sets that we've integrated, and also the contribution by platform, tissue state, whether there are cells and nuclei, and the biological sex of the samples that were analyzed. And you can see the distinctive feature is that the vast majority of the cells are fresh. The vast majority of what we have are from cells rather than nuclei, but a reasonable distribution across sex. And this is what the integrated object looks like, which has been visualized and pseudo-colored based on organ of contribution and also by gestational stage. And the big section here, which is in light blue, represents the brain, which is the biggest, contributing the biggest number of cells in the embryo during this developmental stage, but also within the data set that has been captured. But how do we know that this is a really valuable and robust integration, given that there's a lot of biological differences from the multiple organs and the gestational time, and the fear of any integration is that one throws the baby out with the bathwater in trying to integrate and actually lose the actual biology that we really care for. So we looked at several integration assessments, those that have focused on batch effects, but also those that are looking at biological conservation, including some new methods, which include trajectory-based metrics. And you can now display those trajectory metrics, which are pseudotime, embedding, and adjacency-based, alongside the more recently developed SCIB metrics, and provide an aggregate score, which allows you to assess the performance of the integration. And largely, we found ScanV to be the method of choice for the integration efforts of developmental data sets. Now that we have an integrated object, the next stage will be to look at the annotation or the cell identity based on gene expression, and there are many factors to consider. We retain the author annotation so that we can actually follow through what the authors originally annotated, and then to speed things up, did a semi-automated using cell typists to get a round of annotations, and then went to more focused community-based annotation, which was actually done in several stages. Firstly, engaging experts in the domains or in those organs, and particularly from the developmental biology community, and then opening up to a wider global community. And I'll put up a QR code to also invite you to participate, so please have your handphones, smartphones ready. So these are several of the names of individuals who've been involved in the community annotation, and I've highlighted the way that we've used cell typists initially, and then using cell by gene portals to get more in-depth input, and then subsequently the data is now available on the cell annotation platform for anyone to provide further feedback. So do feel free to scan the QR code, and you can then sign up to join and give us feedback on the current annotation of the developmental data sets. So what do we really want to do from the developmental integration, the first version of the atlas? The first is to uncover new biology. What do you see when you bring together all of the different organs? One fantastic example that came out from this effort was the ability to annotate the developing peripheral nervous system from neural crest formation all the way to the formation of the sensory neurons and the enteric, the parasympathetic, and also the glial cells. This was only evident from the integration and was impossible to appreciate based on individual organs. Here's where a distributed network really comes to life from the integration. Another value and utility of the atlas is in being able to bridge across data sets. So for human development, there have been a lot of pre-implantation embryo studies. And then you also have additional developmental data sets that have been generated using completely different platforms. How can the Human Developmental Cell Atlas that we've generated with the community help bridge with the early stage pre-implantation embryo and also be used to provide a model, an SC Archers-based model in this case, that will then help any user to annotate their data set and also to harmonize with existing large-scale data sets. And finally, the value and utility of the developmental atlas from the perspective of clinical application. And here, how the integrated atlas, including particularly whole embryo single-cell RNA sequencing data from one gestational stage, can be used to guide clinicians to look at how the... Is it possible to link to the web link there? If you just click on that, it should take you to a website. I thought there would be a computer screen there, but not. So if you, anyway, it takes you to the Decipher. And what you see on Decipher is essentially how you can visualize where each of these genes that are expressed in the whole embryo, which cells are expressing them, and where in the embryo it is being expressed. And so you can look at, for example, a gene that is known to cause Holt-Oram syndrome, TBX5, and how it's located to the specific parts in the embryo, particularly in the heart, and it's known to cause congenital heart problems. So here you can really begin to understand how developmental disorders manifest during in utero development and what a clinician should be looking out for, and it's directly linked with patient clinical symptoms, and hopefully this will drive forwards more precise ways and also greater insights in terms of what to be looking out for, what management is needed, what investigations are needed, and potentially how these children will be treated and monitored after birth. So I'm going to stop here, and I'm just going to leave that QR code for anyone who hasn't had the opportunity to scan and participate. And thank you very much for listening.
So with that, please join me to thank all of our amazing speakers for this session. I hope you have a better sense of our roadmap and where we're going next. So we were planning on taking questions, but to be honest, we're running a little late, and I wouldn't want you to miss a well-deserved break after so much science and so many talks. And so during this break, which is going to end at 11:20, we ask you to be back at 11:20. For those of you that are interested in meeting with the Nature Method editors, please go to the registration desk. And otherwise, we invite you to browse all the amazing booths we have, a lot of different technologies being showcased by our different sponsors. So do go and wander around, and please be back at 11:20. Thank you. Okay, welcome to this session. Before I introduce our host for this session, I do want to point out that we realized that there was no time for question, but I wanted to highlight that there are several breakout sessions. And these breakout sessions are meant for you to bring your questions, bring your thoughts on different topics, and some of these breakout sessions are going to be highlighted today, so there's going to be plenty of time to have different conversation. We also invite you to track speakers during the different breaks to ask specific questions. And so it is my great pleasure to introduce a colleague and friend, Holger Heyn, who's going to share the next session. Holger is a professor, I'm going to try this in Spanish, Holger can make fun of me, at the Centro Nacional de Análisis Genómico, or the National Center for Genomic Analysis. I know, I don't speak Spanish. Holger also wears many hats for HCA. He's the co-chair of the Standard Technology Working Group. He also co-chairs our new industry program partnerships. And together with Shyam, Muz, and myself, the four of us, have been working on the spatial strategic planning. And so Holger will give you an overview of what we've been cooking.
It could have been much easier. So I'm group leader at the Spanish Genome Center in Barcelona. That's a much easier way to look at it. So welcome everyone back from the coffee break. I hope you're sufficiently caffeinated, especially my European colleagues, because for us it's very early. So it's a pleasure to be here, and it's a pleasure to be here as a co-chair of the Spatial Task Force, because that's my major function today, chairing this session, not only having excellent scientific talks, but also giving a bit of an introduction about the Spatial Task Force and the activities we have been performing over the last year, because last year at the meeting in Milan, at the annual meeting, we have been launching the Spatial program, which is really meant to bring the human cell atlas into the next phase. So moving away from building atlases with dissociated cells, but really to map cells in their natural environment, being a tissue, organs, to really use then spatial genomics technologies that we now deem fit, really to generate atlases at high quality at scale. So technologies evolved over the last years, and now we're launching the next phase using those technologies and building spatial atlases at scale. So this is an enormous task, and we broke it down in several tracks. So these tracks are organized by different leads and kind of chairing different teams. And the really goal is to define initially critical questions, challenges, and opportunities to build such spatial program. But then really the output should be informing a strategic plan moving forward, building a roadmap towards building spatially resolved atlases at scale across all bionetworks eventually. So I had the pleasure to co-lead the track 1, focusing on technology, together with Shin. And the different tracks were focusing on computational and algorithm development with track 2, portal development track 3, and the track 4 with Chloe Villani, then bringing it all together in using the kind of expertise from the different tracks towards launching a atlas generation phase, building spatial atlases, not only in health, but also in the disease context. And my task for the next now 7 minutes or so is really to report back what we have been discussing over the last year, but also to kind of bring you the roadmap towards building such atlases in the next 5 to 10 years. So focusing first on the technology part, I had the pleasure to kind of bring a group of experts that were not only kind of experts in the technology themselves, but also were very experienced using those technologies not only in atlas building efforts, but also in multiple disease contexts. And initially, we have been mapping the technology landscape. So what is out there, technologies that would allow us building a spatially resolved atlas. And we looked at key features initially, not only how these what is inherent technology behind those methods, either working on capture per sequencing based atlas building or spatial atlas building or imaging based methods, but really then kind of stress tested those methods for their ability to build spatial atlases at scale. So we were looking at area size, so what size of a tissue we can capture with the respective technologies, but also what can be used as input material, for example, FFP or fresh frozen material. A large discussion that kind of was discussed across all tracks was the building of marker panels, going beyond potentially an exploratory phase where we use transcriptome-wide or even transcriptome scale methods on the imaging side or on the sequencing side, but then use that experience plus the experience from our version 1 dissociation cell-based atlases to generate marker panels that might inform us and allow us to kind of scale cost efficiently with high data quality across many organs in the end, but also scaling towards different disease contexts. Building atlases at scale needs a high or quick turnaround times. We were looking at this, but also the performance across different organs, because not all technologies would work sufficiently well across all organ systems. At this point, we are focusing on the 2D context, but we kind of tackle it already by working on sequential or serial sections to have a 3D component in the design, but eventually we're already kind of scouting the market for 3D technologies being implemented during the roadmap. So with this, we then came up with a short list of technologies that might be suitable to generate those atlases. We matched those to different bionetworks, looking at the best fit for purpose to build atlases across organs and tissues. And we are already in contact with the biotech partners that provide those technologies and discuss how we can team up and collaborate to build those atlases beyond an initial pilot phase towards flagship projects that generate atlases at scale. So track 2, from the computational and algorithm development side, the initial discussion was around the challenges for algorithmic and data analysis, really to empower HCA spatial atlases of healthy tissues, disease, and diversity. So the complexity is increasing. And here, step 1 is basically setting up the subsequent steps for success by building a toolbox around the analysis framework. Again, looking at panel designs. So panel designs is one of the core discussion topics and one of the core kind of priorities in the initial phase of this task force. But also we look at critical QC metrics, mainly around cell segmentation, because in the end, working on dissociated cells, those cell boundaries are clearly defined by the dissociation step. But now looking at cells in space and with the goal really to do single cell profiling in the spatial context. Cell segmentation is one of the most important problems or challenges to figure out. Eventually, we want to move towards the integration of single cell atlases of version 1, cells put in the spatial context, and their method development will be part of the first priorities. And obviously, we'll generate a lot of data, so this has to be managed at scale. So step 1 is basically setting up step 2 for success. Step 2 is then really focusing on the toolbox behind analyzing cells separately, cell-cell combination, cell-cell communication, cells and niches, cell architecture and tissues, integration of multimodal data, integration of germline, and then eventually make things cost-efficient, accessible, potentially even looking at HE and imputation on HE images. In the final step, then, those experiences are taken forward to build cellular or models of cells. In this case, integrating the temporal resolution, so eventually performing spatial temporal modeling, ideally already taking the 3-dimensional context into account. From the portal development, there was an initial strategy to kind of define those spatial web portals, which consisted of the review of existing infrastructure approaches, so infrastructure that has been already set up for raw and processed data, storage, the querying of spatial data, and then the integration with visual inputs for the analysis. And this is now taking into a next step where we will work on use cases and really get the user feedback, how users navigate with the metadata, because now we have a different component, which are cells in space. So we will kind of work very close to the community and the feedback when navigating in space, but then also when navigating across the diversity of cells, looking at cell types and markers. And this is now being assembled as an action roadmap. Track 4, so the track that brings it all together. the one track that rules them all, led by Chloe and all bionetwork coordinators in the end. So thanks for all your input over the last year. Initially, really to define the scope. So the scope of a minimum viable product, the MVP. So what is the endpoint of the process? Why do we do all this? So here the MVP has to be informative. So it has to kind of generate a spatial map of human systems, but it also it has to be actionable. So we should be able at the end of the process to extend, especially in the disease context, the extraction of biomarkers, even the identification of drug targets. So this is kind of at the end of the process, and within the roadmap, obviously there's a big discussion, a priority on metadata, as we have been focusing our metadata approaches on the single cell side. Spatial is dealing with different metadata types, but also with additional measurements. So we're not looking at a single modality, but crucial modalities like H&E or protein that then would allow us to scale cost efficiently across a community and disease types. But also with protein, we are aiming at a very close link to already established pipelines, especially in the clinical context, where protein has been used over the last decades in the end as a main readout for diagnostic and therapeutic purposes. So strategy for building marker panels. So here it's, again, us leveraging our experience from the version 1 atlases. So here we can extract marker panels that will allow us to inform about diversity of specific tissues and organ systems. But we can also look at, for example, cell types that are present across tissues and organs, for example, immune cells, stromal cells. So these marker panels will then allow us to not only dive deep into the complexity of single organs, but also to integrate across organs. And this is currently being discussed with company partners. So we're discussing the generation of those panels, but also the generation of large data sets. In the first phase, potentially a deep dive, as it has been done for the brain, for few samples, but then also to scale this to the disease context and across many more samples using marker panel designs. And with this, I will wrap up this framing talk with a kind of roadmap towards the MVP, where we already started pilot projects. These are really meant, especially for new technologies that are in the market, that are already launched or are being launched pretty soon, to test the scalability of these methods, really towards the generation of 2- and 3-dimensional atlases. Because we kind of shortlisted key organs, where we're now working on few donors, series sections, to integrate those into high-quality draft atlases, which will then inform a next phase where we work on flagship projects. So flagship projects are really meant to map bionetworks, organs, tissues at scale. So here we're looking at multiple donors, multiple sites, really a phase that allows us to move beyond towards scaling across organs and tissues, so to get a comprehensive insight about the complexity of full tissues, but then also scaling across populations, eventually to define that minimal viable product that allows us to assess utility being informative and actionable. So over the last year, we haven't been working in isolation, but we've been interacting with the rest of the HCA community through surveys, and one of these surveys was focusing on the spatial context, and we're very pleased to see that many of you already are generating spatial genomics data in one way or the other, and also that they are whole organs and larger, let's see if that works. Yeah, so that we have whole organs, but also larger sections or resections available from surgery or autopsies that will allow us to be used, or that can be used as an input material to generate those atlases across full organs at scale. And with this, I will finish pointing you to the breakout sessions that we have today, 2 at 5:20 on spatial technologies and spatial computational tool development, and 1 tomorrow focusing on the portal site. And with this, I will finish and start the scientific part of this session. And it's a pleasure to introduce Stefania Giacomello from the KTH in Stockholm in Sweden. And she will talk about the human and not so human part of spatial transcriptomic analysis. Welcome.
Good morning, everyone, and thanks to the organizer for inviting me to this very great meeting to present us what we are doing in the lab. In the lab, we develop and apply technologies in order to study the spatial organization of gene expression patterns, and we do that across kingdoms, specifically across animals and plants, which both kingdoms actually share the fundamental principles that from cells, single cells, the fundamental unit of life, they arrange themselves spatially in order to generate tissues that they assemble themselves into systems, and then from systems you can obtain functional organisms. And here is an example, actually, of how preserved gene expression patterns are in different kingdoms. I'm sure you know very well the preservation across the animal kingdom, but also, as you see it on the right side, these preservations of spatial patterns of gene expression occurs in plants as well. Recently, we have become very interested in the following key questions. How do infection processes alter the host spatial gene expression? And also, what are the crosstalks between the pathogen and the host? In the lab, we work with the spatial transcriptomics technology that was developed at SciLife Lab in 2016, which nowadays is known as the Visium technology distributed by Teichmann Genomics. Probably you already know that the technology relies on glass slides, as you see it here, which is divided into capture areas, and in these capture areas you have spots which contain barcoded poly-DT probes. By having only poly-DT probes on the surface, if you are interested in studying microbial patterns, you encounter a severe limitation since microbial RNA expression is usually not polyadenylated. To overcome this challenge, we have been developing spatial metatranscriptomics, which relies, again, on glass slides, which is divided into capture areas, and these capture areas are also arranged into spots of 55 micrometer in diameter. The main difference here is that probes on the surface not only contain a poly-DT stretch, but also contain specific probes for 16S molecules and also 18S molecules as well, that you can play around with different type of proportions in terms of concentrations. A workflow starts by attaching a tissue section onto these capture areas. You perform a permeabilization that is both for the host and the microbial information, and at that point the RNA flows down to the surface and get captures from the different type of probes that we have. Afterwards, the workflow is pretty much as the current Visium workflow that you already know. A main difference is how you handle the data, because at this point you need to align your reads not only to the host genome reference, but also to microbial data sets in order to extract 3 different types of readouts in the end, which would be the bacterial readout, the fungal readout, as well as the host readout. Once you have these 3 different readouts, you can do a very wide set of type of analysis. For example, you can look into the microbial geography, so where actually bacteria and fungi, for example, are expressed or are present on a tissue section, and if they, for example, arrange themselves into hotspots. So hotspots are essentially parts of the tissues that are significantly enriched for the presence of microbes. You can also perform interactomes analysis, which essentially means looking for specific interactions between bacteria and fungi. And also you can perform the more standard type of analysis on the host response. Okay, yep. We are applying this technology also in the animal kingdom, specifically to generate a functional cartography of intestinal host microbiome interactions. And we are interested actually in studying these host microbiome interactions when individuals are affected by inflammatory bowel disease, so IBD, or colorectal cancer, CRC. And in general, what we are trying to understand if is during these diseases, there are like different type of niches of host microbial interactions in a normal part of the tissue, so not disease affected, and in a disease affected part of the tissue. In this case, of course, we are looking at the colon tissue samples. The overall idea is then to understand if there are like specific biomarkers, both at the human or also bacterial level, that can kind of distinct, these different niches of interactions. And here is an example on a mouse colon tissue section that was analyzed with SMT. So on the first panel on the left side, you see the distribution of the host reads that exactly match the host tissue morphology, so the mucosal layer. In the middle, you have a distribution of the bacterial reads that are specifically spatially mapped to the stool pellet. In the third panel, you start seeing a combination of different type of clustering analysis that you can perform by having these different type of readouts. And in the last one, you can see that there is a specific activation of the inflammatory JAK-STAT pathway in the intersection layer between the mucosa and the stool pellet, which actually make kind of sense because in IBD, usually you treat inflammation with inhibitors of the JAK-STAT pathway. So essentially to say that with this method, we are really able to see kind of key signals and that can lead us, for example, in the future to identify biomarkers. Now in the last part of the talk, coming to our latest technology developments that are ongoing, we are combining spatial metatranscriptomics together with spatial VDJ that you probably heard of, which is essentially a technology that allows you to follow the spatial distribution of B cells and T cells in the tissue sections, and also how these immune clones they are spatially arranged in the section. In this case, we are working with the human tonsil, and we take tissue sections from the sample, and by attaching it onto a SMT slide, we can obtain both the microbial information as well as the spatial VDJ or clonal information. And at that point, we can start looking at host microbiome interactions. Here we can see the bacterial enrichment in 4 different tissue sections of human tonsil, and as you can see, the spatial the bacterial distribution is fairly consistent across the 4 tissue sections, as well as the 4 top most abundant bacteria are in agreement with the topmost 4 abundant bacteria that were identified in a tonsil sample for 16S sequencing. You can also spatially map, as I was saying, all the bacterial information. So here again, you see the 4 top bacteria that are identified in our samples. And also you can start looking into these different type of interactions. So for example, on the left side, you see the tissue morphology and the host clustering analysis that we have performed. And we obtain 8 different clusters that correspond to the expected anatomy of human tonsil. On the top right panel, instead, you see the bacterial distribution in the tissue sections, and you can see that there are like some specific enriched areas of bacterial content, which actually corresponds to the cluster 4 and 5 in the tonsil anatomy that are specific for the epithelial tissue and for the crypts, which the crypt is a part of the tonsil that creates an invaginations where essentially bacteria are in contact with the organ. Moreover, you can also follow the distribution of the different immune clones, of the human immune clones. And you can see that we have like overall a widespread distribution across the tissue, but most of them are localized again into the top 3 clusters, so specific for tissues that contain T cells and B cells. and also for cluster 6 and 7, specific for plasma cells. Now, looking at the general overlap between the bacterial distribution and the clonal distribution, if we focus in this area here, you see that we have a higher bacterial density in the crypt, and there is also a higher clonal density around those crypts. And here essentially is like a visual representation of what I was describing. So essentially you see that bacteria are specifically localized in this portion of the tissue, and overall we have clones all around it. So what we are interested in understanding is actually why we see these spatial patterns, and also what is in this kind of interface layer between the bacteria and the clones. Finally, what you can also do with this type of information is, for example, to follow at this point the distribution of bacterial or clonal enrichment based on distance from the clones, from the clusters of interest. So here, again, you have the bacterial enrichment, and if we take this one as an edge, as a border, essentially, of the cluster, you can see that from the middle of it, you have a much higher bacterial density that slowly decreases moving out from the crypt, while the clonal information, the clonal amount is much lower in the middle and increases moving out from it. So with that, I think I have I ran out of 10 minutes, so I will skip this one. But essentially just to say that we are also extensively working on applications of these methods, both to mouse brain affected by spaceflight and the development of plant organs, specifically Norway spruce. To conclude, I have this kind of very slim slide with only 3 points, but actually summarizes years of work that we are doing in the lab, which essentially we are very focused on extending SMT for FFP samples, since the current technology is applicable only to fresh frozen tissue samples, and also to achieve the microbial taxonomical resolution of the method. With that, I would like to thank all my lab members, very talented people that have been working on the development of these methods, all our collaborators, funding agencies, also 10x Genomics for their support, and you for the attention.
Thank you very much. Very cool talk, very cool technology. We have time for one question. Must be up there. Huh?
Thanks, Evan Keller from University of Michigan. Wonderful talk. Quick question: when you think about a 55 micron spot, there's probably 1,000 to 4,000 bacteria in that one spot, right? How do you deconvolve all the different species of bacteria in there to find out what you're really dealing with?
Yeah, thanks for the question. That's exactly what I was leading to in my conclusion slide. So at the moment, within 55 micrometers, it's difficult to identify all the bacteria, plus the method at the moment stops at the genus level for taxonomical classification, so we want to push it to the species level. Then at that point, you really see at the spatial distribution of each single bacteria. At that point, you really need to perform some imaging type of approaches, which in our group for carto-host-back we do have, and are like single molecule fish type of approaches.
So thank you very much. Big applause again for Stefania. And next on stage, we have Mingyao Li, professor at the University of Pennsylvania, talking about the smart AI-based analysis of spatial data. Welcome.
Waiting for my slides to show up. Hi, good morning, everyone. I want to thank the meeting organizers for inviting me to speak. I'm very glad to have this opportunity to present my work on how to use AI to accelerate spatial omics research. So single-cell sequencing is a commonly used approach to study the molecular behavior of cells in multicellular organisms like human. But more recently, newer technologies like spatial omics have gained more popularity. As compared to single-cell sequencing, spatial omics allows us to measure the molecular information of each individual cell in their original tissue context. And such spatial location information is essential for understanding how the tissue is spatially organized and how the cells communicate with each other. So there has been many platforms that developed for spatial omics, and here I list some of the popular ones that have been commercialized. Although these platforms are different in terms of their spatial resolution and the molecular content they measure, there is one thing that is shared in common. That is, they are all extremely expensive. And such high cost has prevented the broad applications of spatial omics technologies in large-scale tissue-based studies, such as the Human Cell Atlas. But interestingly, many of the spatial omics technologies allow the generation of a paired histology image from the same tissue slice from where the molecular data is obtained. And oftentimes, these histology images are high resolution, extremely high resolution indeed, and more importantly, they are very cheap and easy to generate. So this is in sharp contrast to this expensive spatial omics data that we generate. So the main focus of my research in the last couple of years has been trying to develop machine learning and AI tools so that we can combine the strength of spatial omics and the pathology imaging, overcome their individual limitations so that we can get the most out of a spatial omics experiment and achieve scalability, accessibility, and speed. So first, I'm going to demonstrate to you how AI can leverage high-resolution histology image information to enhance the spatial resolution of gene expression data generated from a platform like 10x Visium. So Visium is a very popular platform. You heard it from the previous speaker's presentation. And indeed, a large amount of data has already been generated using Visium. But a key limitation of Visium is the lack of a single-cell resolution. And this makes it difficult to characterize both the within spot and between spot spatial structure of the tissue that we care about. But an advantage of the Visium platform is that it allows the generation of a high-resolution histology image before the actual Visium experiment. And this histology image information provides extremely valuable information, especially when we study human disease. And the other thing that we noticed when working with numerous amounts of spatial transcriptomics data is that typically similar histological patterns imply similar gene expression profiles. So this means that if we can systematically learn the relationships between histological features and the gene expression patterns, then there should be a possibility for us to use the histology image information to enhance the spatial resolution of gene expression within the directly measured spots. And furthermore, we can leverage the high-resolution histology image information to predict the missing gene expression in the tissue gaps with high resolution. The reason is that the histology image does not have any tissue gaps. So motivated by this, we recently developed a tool called iStar, which is able to use AI to enhance the spatial resolution of gene expression data generated from a platform like Visium by utilizing the high-resolution information provided by the cheap histology image information. And iStar contains multiple components, including how to hierarchically tell a high-resolution histology image, how to hierarchically extract the image features, and how to use the extracted image features to enhance the spatial resolution of gene expression, and then finally, how to annotate the tissue architecture at high resolution. So the most important step in iStar is to use a hierarchical vision transformer to hierarchically extract the information out of a histology image. And using this approach, we are able to extract the high-level image features that reflect the global tissue architecture. And we are also able to extract the low-level image features that are reflecting the local tissue cellular structure. And this is exactly mimicking how a pathologist visually examines a histology image under the microscope. And once these hierarchically extracted image features are obtained, what we do next is to build a weakly supervised machine learning model and virtually predict the super resolution gene expression data for each of the single gene in the genome. And here, the weak supervision is provided by the low resolution spot level gene expression. So by the end of this analysis, we are able to turn a low resolution gene expression, like what you see in the middle, into a high resolution gene expression that achieves single cell resolution, like what you see on the right. So this is like you originally took a picture using your iPhone, but now using super resolution AI technology, you are able to turn your low resolution image into a high resolution image, as if you have taken it using a professional grade camera. And this is exactly what we do with the iStar technology. So iStar is developed for a single sample analysis. But as you have heard from an earlier talk in the morning, all of the existing spatial omics technologies have a size limitation in terms of the tissue capture area that they can measure. I wanted to mention that iStar can be applied to many different tasks. So for example, using the super resolution gene expression data we obtain from iStar, we can segment the tissue at very high resolution. And this allows us to obtain very fine-grained, detailed structure information like the intratumoral heterogeneity. And that is not possible when you use the original low-resolution spot level gene expression. And furthermore, we can annotate the cell types at single cell resolution, and this provides valuable information about the high resolution tissue organization and the disease microenvironments. And now I'm going to move on to how to scale up spatial transcriptomics to large-sized tissue that is beyond the tissue capture area of a traditional spatial omics platform. As we have heard in the morning, many of the human tissues, especially human brains, are very large in terms of the physical size. They are much larger than what the existing platform can directly measure. So a natural study design is to use this grid layout, where we cut the tissue into smaller pieces following this grid, and then measure each of the small pieces using an existing platform. But in reality, this grid layout design is very hard to implement because each tissue section is very thin. So in order to get enough representations, a researcher may have to cut multiple consecutive tissue sections so that we can get enough data captures to measure and quantify the molecular diversity in the original tissue block. So in the end, we will get a set of smaller tissue pieces. It's like you have a set of puzzles. And now the task is how to reassemble them back to the original tissue space. So to solve this challenge, we recently developed a tool called iScale, where we can scale up spatial transcriptomics to any large-sized tissue that is beyond the traditional spatial omics platform, as long as you can generate a high-resolution histology image from the tissue that you wanted to study. And here, we leveraged this multi-capture study design I introduced in the previous slide. And we have a procedure that can semi-automatically align the data captures back onto the mother image, which characterizes the large piece of tissue that we wanted to study. And after that, we have a data integration procedure that allows us to integrate all of the gene expression information together. And then we can also use some machine learning to learn the relationships between histological features and the gene expression patterns. And this allows us to virtually broadcast single cell resolution gene expression to the entire large mother image. So we applied iScale to the data generated from a human-based multiple sclerosis study. As you can see, this image that you see on the left is very big. It's 3 centimeters by 3 centimeters, much larger than the Visium platform can directly measure. So in order to study the gene expression variation across the big tissue section, we generated multiple data captures using the multi-section study design I introduced earlier. And here's the data that we obtained. This is like a set of puzzles. And now using iScale, the algorithm we introduced, we are able to map these data captures back onto the 2D space defined by the mother image. And we can further use the I-scale to virtually broadcast the gene expression to the rest of the tissue and generate a cell typing map that has a single cell resolution. So by the end of this analysis, we are able to detect multiple cell populations of microglia around the lesion border that is not possible to detect when you utilize the originally obtained low-resolution fragmented Visium data. And now, in the last part of my talk, I'm going to touch upon the study design problem in spatial omics, which is extremely important but often looked. So when you plan a spatial omics experiment, especially when the tissue is large, you have to decide the optimal region of interest for your experiment. But oftentimes, people just use a visual inspection to select the ROI. And this is subjective to human errors and is not reproducible. So to solve this challenge, we recently developed a tool called S2Omics, standing for Smart Spatial Omics. We aim to use AI to fully automate this process and make this procedure fully reproducible. The idea is to use AI to process and analyze a histology image from the tissue section that you are interested in studying, and then use computational algorithms to select the best area that capture the molecular diversity in the tissue that you wanted to study. So by the end of this analysis, you can follow the ROI we recommend and design your own spatial omics experiment. To see this work in action, we applied S2Omics to a data set generated from a gastric cancer sample, which was originally measured using the expensive Xenium platform, which costs us almost $8,000. So now I wanted to see if the ROI selection can save the experimental cost. You can see that using S2Omics, we can select a very small tissue capillary area, 6 millimeters by 6 millimeters. And this small capillary area captured most of the important tissue structure that are present in the tissue. And to further verify the region we selected is representative, we further broadcasted the information captured in the ROI to the entire tissue section. And you can see we are able to recover the TLS, the tertiary lymphoid structures that are not directly measured in the entire tissue section. So to summarize, spatial omics is a very fast developing field. And in this talk, I have demonstrated to you how AI can accelerate spatial omics research. And as the field continues to advance, I can envision that in the near future, AI is going to play an even more important role, not only in integrating multi-model spatial omics, but also in generating and reconstructing 3D tissue structures and volumes, and also linking the spatial omics data with clinical information to build prediction models for precision medicine. So with that, I'm going to conclude my talk, and I wanted to thank the following members of my lab who contributed to the work that I presented here today. And also I want to thank my collaborators and my funding support. Thank you very much.
Thank you very much. Unfortunately, we do not have time for questions, so we just move directly to the next talk. Presented by Zhenyu Xiao, Associate Professor at the Beijing Institute of Technology, and she will be talking about spatial technologies in early embryonic development.
Okay, thank you for the introduction. And also many thanks for the organizer to invite me here. I'm very small, so I will like this. So, yeah, my name is Zhenyu Xiao from Beijing Institute of Technology. I just started my own lab back in 2022. And today is really my great pleasure to be here to share you with some published and unpublished work recently done in my lab. And so as we can all agree that having a baby should be one of the most fulfilling moments in life. However, this is not always the case because as you can see that even for a very young, healthy couple, they can only got 40% of chances of getting pregnant. And still globally, almost 20% couples are infected by the infertility, a very striking high proportion. And most of those pregnancy loss happened very early at pregnancy, before or during implantation, a stage that are still in the black box. And even after successful implantation, we still have almost 60% of chances of getting a born baby with birth defect. And also for the pregnant mama, life is not that easier. They're faced with numerous pregnancy-related diseases, which significantly affect the quality of their life. So we could partially link those mentioned problems to a very unknown but very important organ, the human placenta. And as we all know that the placenta and also the baby, they are both come from the fertilized egg, but their lifespan are quite different. The placenta can only survive for 10 months. However, it is this 10 months lifetime that can not only support the health of the mother and its baby during pregnancy, but also the lifelong health of the mother and its offspring after birth. So sitting in the maternal-fetal interface, the placenta venous is the functional unit of the placenta that measure two types of cell types that consist within this interface, the trophoblast lineage and the non-trophoblast lineage. And also for trophoblast lineage, there are two mononuclear cell types, the cytotrophoblast cells that can serve as stem cell-like cells, which can differentiate through EMT to form these very invasive EVTs. And also two types of multinuclear cell type, the human STP syncytial trophoblast that forms the out layer of the placenta villi, and also another very unknown syncytial, which is the placenta trophoblast bed joint cells. And since I started my own lab, I was quite fascinated by the cell types within the venous core, which consist of the endothelial cells and also some tissue-resident macrophage, which in the placenta we call Hofbauer cells. So, of course, my first interest goes into this amazing syncytial, which is the STB. According to research, it will contain 58 billion nuclei within one cytoplasm at term. And thanks for the single-cell multi-omics, we can now draw this very beautiful dot painting in the lab with our own computer. So to question the heterogeneity of the nuclei within the STB, and also to see whether those 58 billion nuclei within one cytoplasm that follow this one single differentiation path or not. We first focused our study on the very earliest stage of pregnancy, because a lot of pregnancy loss and also birth defect rooted at this specific earlier stage. And to our surprise, at least those nuclei within the STB, they at least can divide it into three types with a specific gene expression of PAPBA and FLT1. And of course, the immunofluorescence confirmed the existence of these three major STB nuclear cell type. And to see whether they follow this one single differentiation path, the answer is no. We used several analytic methods, and the result turned out to be that they follow this bifurcated differentiation path, and with, of course, specific functions. Like one group of them are actually response to hypoxia, because we know that at early pregnancy, we need this hypoxia environment to really support the vasculation and angiogenesis. And another group goes into the function that are related to the hormone secretion. And with the power of the integration of single nuclear ataxic and single nuclear ion-seq, we had those very beautiful and unique accessibility regions that are responsible for the different differentiation paths of the STB nuclei. And also we identified several low-vol master regulators with their downstream regulated genes. And of course, in the lab, we are very happy with those enhanced gene regulatory network. We further confirmed the master regulators with a coexistence of the master regulators together with its regulated genes. And then, because we also constructed this human
trophoblast stem cells and also trophoblast placenta organoid in lab. Then next, we questioned to how much extent our mini placenta in the lab or placenta organoid can mimic those in vivo data sets. Of course, when we integrated the data sets from in vitro cultured placenta organoid with the in vivo ones, we can see that the major mark gene expression are quite overlap with each other. However, no matter what method we used in the lab, our mini placenta in the lab can not mimic those very matured functional STB nuclei in vitro. So with this master regulator we identified in the lab or in our data sets, we CRISPR-Cas those master regulators, and now we can construct a more bona fide placenta organoid in the lab, and also we can use them to really monitor those pregnancy-related diseases and test a lot of drugs. So at the end of this project, I also started my lab. I was fascinated again with the human early embryonic development. So with my former PhD professor supervisor Susanna in LMC, we collected several very precious human embryos at very early stage under very strict ethical regulations. And we now could not only draw this nice placenta atlas, but also we can draw cell atlas about the embryos and also another unknown organ, which is the yolk sac. So we all know that the very early third week, a lot of events in this week are still in the black box. So to shed light on those very early stage of human development, we focused our study on the ankle formation with a focus on the node and the notochord, because we all know that we are not born with our ankles like anterior, posterior, left, right, and dorsal and ventral. So we need an organizer just like Teichmann in Beijing to tell us the ankles. So we employed the spatial transcriptomic, and also we sequenced the 62 transverse section of the human Carnegie stage 8 embryos, and together with all these slices, we used paste method to construct this human Carnegie stage 8 embryos in the lab. And then with this 3D atlas, we want to first focus our study on the specification of the mesoderm cells. So as we all know that in the in vitro model, people proposed that mesoderm cells is already specified along the primitive streak during gastrulation, and our result further confirmed the theory, the hypothesis that a lot of mesoderm is already specified along the primitive streak, like the axial mesoderm, paramesoderm, intermesoderm, and the lateral plate mesoderm. And also, we are focused our study on the notochord, and to our surprise, we not only identified the notochord that are responsible for the ankle formation, but also we identified another lower organizers, which is the collecting stalk. And the collecting stalk will finally develop into the umbilical cord at a later stage of human development. And then to question how conserve between mouse and human organizers, we moved our study into an earlier stage, human embryos, which is the Carnegie stage 7. We also identified the AVE-like hypoblast in the human embryos. And at the same time, we identified another amazing organizer, which is the node. And we proposed that node can not only derived from gastrulation, but before gastrulation, they might come from directly from the epiblast. And then we focused our study on a later stage of human embryos at the end of gastrulation and also at the beginning of organogenesis. And we proposed that the NMP, neuromesodermal progenitor, that actually form these two-layer patterns along the DV axis to form the somates and also the spinal cord and the part of the hindbrain. And now we are constructing those the formation of the heart and
and also the inner ear in the lab. And as you can see in the movie, that we finally successfully constructed the very amazing cochlea and vestibular system in the lab. And hopefully we can report this result to you in more detail next year. And the manuscript is under preparation. So because of time limitation, I will stop my presentation here. And many thanks to the organizer, to my PhD supervisor, and to my very talented students in BIT. Thank you very much.
Thank you very much. For the sake of time, we skipped questions and bring these questions to the coffee break and lunch break. Next on stage, the one and only Arutha Kulasinghe from Queensland University, talking about the clinical application of spatial genomics.
Thank you, Holger, and it's great to be at this meeting. Can you give me a wave if you can hear me out back? All good, thank you. So let's just stick to time, try to stick to time. Holger, I've got 10, so let's go. So I'm acknowledging a number of funding agencies that support this work. I'm based at the Fraser Institute at the University of Queensland. So it took Eric Topol to define this field, and it was really interesting. He called it spatial medicine. And it's really this evolution of technologies coming to a precipice where we can really apply them across clinical cohort studies. Nature called this tumor geography. There are multiple different terms that people are using to really describe these, but I think spatial medicine is where this field is really heading, and we're super excited by it. The work that we do is trying to really understand what's going on in the tumor microenvironment in cancer, looking at predictive biomarkers for checkpoint inhibitor therapies. A lot of studies you'll see out there are single site studies, single hospital studies. The challenge in the field is to develop a multi-marker signature that you can really drive into companion diagnostic assays. And so what my lab tries to do is to map every cell within a patient's biopsy or surgical resection, 3 to 5 million cells per patient per sample. These are some of the ongoing studies in the lab in head and neck, lung, breast, colorectal cancer, and we've got pandemic studies looking at the impact of viral infection across multiple different organs, but also contrasting across the adults versus children. I'll focus on the head and neck and lung studies, which have significant numbers, both retrospective and prospective studies, and tapping into clinical trials as well. So we do spatial proteomics and transcriptomics. I had a slide on the technologies we use, but I've pulled it out in the interest of time. This is a single cell proteomic pipeline that we now apply on all our workflows, in addition to whole transcriptome, single cell and subcellular. We use a flavor of metabolic and functional insights to subset within each of the cell types, and we've got a number of papers I've flagged there. This is a study that's currently under review, but I'll quickly go through it. This is a non-small cell lung cancer single agent IO up front, where we ran this pheноcyclofusion technology, which is single cell proteomics, where we built in these functional and metabolic insights into the cell phenotyping. So at a sort of a category level, you're not going to really see it. You really need to go into the subsets. And so we had clinical endpoints. This is a study cohort breakdown of each of the individual categories. We had best objective response rate, and clinical benefit at 6 months. And so we can group these by responder, non-responder, but we can break this down here at the tumor stroma level by composition, but then break it down further by functional layers on global counts, but also clinical benefit at 6 months on single agent immunotherapy. And so you can see where the stars are where you've got significance, and you can see this B cell population that is glucose 6-phosphate dehydrogenase, G6PD high, is associated with a better outcome. And so we really wanted to drill into where this signal is coming from. So we split this across this K2, K3 axis, which is tumor stroma and tumor stroma neighborhood, tumor stroma interface. And so your interface or your battle zones are where this action really happens, right? So we can start to see that B cell population, which is metabolically profiled, is actually coming out in that interface between the tumor and stroma in that tissue. And so we use this now to overlay metabolic information, so we can break this down into high metabolic, low metabolic, and intermediate metabolic regions, and we can map that signal directly back onto the tissue. So not just phenotype, but metabolic information directly over the tissue. And so we have the clinical endpoints, so we know which patients had stable disease, partial response, progressive disease, and complete response against RECIST 1.1. And so we can start to look at the cellular sort of metabolic neighborhoods that associate with these clinical endpoints. You can see the activity zones are really in the central area. I'm not going to go into each breakdown here, but you can see the CNs defined by metabolic neighborhoods in there, and you can see the clinical endpoints at the bottom. And so one of the challenges with spatial analysis is how do you use all these different proximity, entropy, Shannon diversity index, neighborhood analyses in a model to really understand and tease out biomarkers that are associated with clinical endpoints. And so Aaron in the lab, working off a Nature Biotechnology paper, he pulled in all G-cross cell proportions, Shannon diversity index, and pushed it through the stable pipeline that can rank your features by your FDR cut point. And so you can start to tease out, using function, metabolic, and phenotype level information, what are those significant biomarkers that associate with your clinical endpoints. This is taking a snapshot into that, so it's taking all your spatial analysis, if you will, and then pushing it through that model. And now we can start to tease that out. So we can see that for resistance to checkpoint inhibitor therapy, we see this macrophage phenotype, which is in close proximity to granzyme B activity with a high metabolic background. So these are sort of ways in which we can tease apart that single cell protein data to pull out associations with clinical endpoints such as resistance and response at 6 months. And so this is now going into a BMS clinical trial, single agent Nivo versus standard of care, whole tissue. So the previous study was on TMAs. We can map these. This is about 4 to 5 million cells per slide. And so every sample, we're running multiples of these. And so we can now start to contrast tumor stroma interface, metabolic neighborhoods, but also tumor density contour profiles between responders and non-responders. And so this work is still under review, but you can start to see, CD4s in the tumor region with glycolysis high is associated with a certain outcome. LAMP1, HLA-DR positive, PD-L1 positive macrophages are coming out. So you can use these different flavors to really break down the microenvironment. What happens when you look at the RNA level? Contrasting responders to non-responders, now tumor stroma breakdown. So what we find is that in the tumor, we have increased expression of GPX1 and GPX4 driving resistance to immunotherapy. So these are genes associated with ferroptosis and iron-dependent cell death pathway. And so we see this in every contrast. So GPX1 and GPX4 is present in every contrast between every version of CR, PR, SD versus PD, CR versus PD, or CR versus PD. So these are clinical endpoints where we're contrasting response against the pre-treatment specimen. So what does this look like when we look at it at a single cell level? This is a whole tissue from a patient that was resistant to checkpoint inhibitor therapy. So follow this with me. Red is high, blue is low. You have high GPX1 within the microenvironment in the tumor, driven by the tumor, GPX1 and GPX4. This is the tumor. Red is high, blue is low. Your CD8s are on the periphery. They're not infiltrating. Your macrophages are on the periphery. They're not infiltrating. GLUT1, so glucose, is like a gradient-like expression through the tumor cells. G6PD, also a gradient-like expression through the tumor cells. CPT1A, fatty acid metabolism localizing to where the T cells are. So you can see this tumor that has increased expression for these markers associated with ferroptosis, driving resistance, but also now when you layer in the protein data, you can see that the cell types or the infiltration levels are quite low. And so we've built these sort of spatial metabolic maps, and this was interesting because AstraZeneca published a study in Nature 2 years ago, which showed that ferroptosis was needed for the maturation of MDSCs. We see the signal coming from the tumor cells in single-agent IO treated patients. And so one of the challenges for us is, how do you look at receptor-ligand interactions visually? And so we've run this in head and neck. This is using rolling circle amplification between PD-1 and PD-L1. And so we can start to toggle between cell types and PD-1, PD-L1 interactions and also multiplex this. And so in the purple here, we have the PD-1, PD-L1 actual interactions. This is not inferred. This is interactions where you have the receptor and ligand within 40 nanometers of each other, and you get rolling circle amplification to visualize that signal. You can see the clear differences between the complete responder and the progressive disease. The mixed response groups, the purple is the PD-1, PD-L1 interaction. The mixed groups are the PR and the SDs here. And so we can look at this in skin cancer as well. Skin is great because we can sample this over the course of immunotherapy. At cure and at progression, we get tissue. And so I won't go into the findings, but we're starting to look at responders and non-responders over the course of checkpoint therapy. At cure and at progression, we get tissue. So 6 time points over the course of therapy. And we can start to see proximity differences between your baseline and your follow-up sample, between progressors and non-progressors. And I'll sort of finish with a couple stories here. This is a whole transcriptome, single cell, 510 sample study where we've looked at tissue arrays, and this was multiple TMAs, about 100 cores per slide. We're getting about 500 to 3,500 genes per cell, but now spatially annotated. So I think we're at equivalency using some of these newer technologies. Super excited by the study. This is just the image of one of those. And so we've got path annotations on the H&Es and so on. I'll skip through this. We've referenced it to the Lung Cancer Reference Atlas. And I'll end with 2 stories. So this is a graph contrast learning model that we've developed on non-small cell lung cancer, early stage lung cancer, resected lung cancer, where we had relapse or no relapse put in as a binary input. This is spatial proteomic data in the middle here showing your cell types. What you have below is a relapse score on your tissue, on your primary tissue. So those are areas associated with relapse that are ranked and shown on your tissue, but based on primary tissue itself. And so I think this is where the spatial data really gets exciting, where you can start to put some of this data back on. And I'll just say, don't throw away your tissues. We'll end with this. This is a 105-year-old Spanish flu study. We've done the RNA and protein, 580-plex protein and whole transcriptome. I won't go into the plots, but that's the DE between 2 of those regions of interest, the RNAs at the bottom here. The takeaway, though, is your PCA is separating out your histology at both the protein and the RNA level. That's what it looks like at the single cell protein level. So I think a lot of these tools can be applied to tissues and archival tissues. And with that, thank you, Holger, for not throwing me off stage.
Thanks, Arutha, for staying perfectly in time. And we're wrapping up now with Yanyi Huang, professor at Peking University, talking about new barcoding schemes in spatial genomics.
Okay, thank you, Holger. I know I'm standing here between you guys on lunch. I don't feel good. So I'll be very quick. So I'm a chemist. So a chemist thinking about the single cell or spatial omics. So we always go back to very, very early stage. So I'm thinking about how to decode or encode all these informations into the reactions. So this one example, I make some toys, like sequencers. So we realized that the most valuable assessment is to calculate how much information you can generate from single reaction. When you mean reaction, you mean either sequencing by synthesis, you generate one base, or you sequence by ligation, you ligate one oligo, or you can do the fish, you do the hybridization. That means the one reaction. So how much information you can generate from this reaction. So to sequencer, you actually squeeze very much. So you try to squeeze out as much information as possible from one single reaction. But when you do this, the spatial optics, we figure out probably not this direction. We can do very luxury things. We can spoil our information. We can actually take so much information we want just to get some more interesting, so much bits to get some more interesting information we want to use for single omics. So I'll talk about these 2 methods we developed. The left part is actually based on in situ sequencing, and right part is based on in situ hybridization. So you have to make some instrument by yourself if you want to do this, or you're rich enough, you can buy it. Yeah. And you actually need to amplify the signal from one particular molecule to amplify it into many, many molecules. So they have the same clonal amplified information, so you can actually extract it much easier. And you need to figure out how to do this. So we use Illumina sequencing reagent, sequenced by synthesis. So we figure out probably we can do them much easier, because at the end, it doesn't really require that many bits to actually encode those information you want to retrieve from the sample. So we use this, we call it the hybrid block code. Only 3 bases be used for coding this sequence, the barcode, and we add one particular constraint, like if any 2 barcodes are overlapped physically in a space, you can actually decode and decouple these 2 barcodes. So this gives you one example that with 10 bases, only 3 bases have been used, and no A, just CGT, right? So only 3 bases be used for coding. And for 10 bases long, this article, you can code about 350 barcodes there. And if any of these two overlapped, you can actually decode these 2. Okay, so this way you can have the subcellular resolution, because you are targeting, using imaging to target every single clone of the amplified gene located in the particular space in the cell, actually 3-dimensionally. But this one I'm talking about is 2-dimensional images. So I'll go very quickly, right? So it's a very super rapid method from sample to 2D images, subcellular resolution, whole clonal section of the mouse brain, less than a day. It takes about like 8 hours to prepare, to do the application, to prepare a sample, and about 8 to 10 hours to take the images, 10 rounds. So essentially you build a poor man's sequencer by yourself and use this sequencing reagent to do the in situ sequencing in the tissue. But people always take this like in situ sequencing against in situ hybridization. However, for me, it's not like this case. I think there's particular 2 categories, 2 kingdoms of this imaging-based spatial omics. One is you need to do cyclic reaction. Cycle by cycle, you react, you take image, you wash it away, you react again. So it's like you put a washing machine with a microscope, right? Or dishwashing machine, better way. You heat it, you wash it, you take a microscope images, you heat it, wash it. But the other category is you don't do fluidics, just reaction and take an image. That's how we do in pathological labs. Mingyao just mentioned it, like every single cell-based whatever omics is so expensive, spatial even more expensive. But pathological labs, all these reactions and the diagnosis are so cheap, right? So I'm asking myself, so what can we do with single reaction, single shot, use conventional available microscope? How many plex? What's the multiplex, the max number you can achieve? Okay. So we have this idea, and we collaborate with Chunhong, we call this a prism. So I just go very quickly. This is one single staining process, and take an image. Use conventional 4-channel microscope, first microscope. Almost every lab can afford this microscope and do the imaging. And we use hybridization because it's a very convenient way, so the pathologist will like it. You can do it in the lab by yourself. So any normal bench work can be justified, just can finish this work in their own lab. So we mix these probes, we tweaked it into a spatial like ratio-based barcoding and do this ratio-based hyperspectrum decoding. So people think it's so easy, right? If you have a 4-channel, you can differentiate this the intensity into different, say 4 grades. Then you have a 4 to the 4th, like 256 barcodes. No, it's not the case because every single spot needs to be amplified. So amplification yield is not controllable. So if a big spot, small spot, you cannot really tell it's because you code it differently or just come from the same gene, but be amplified in the different yield. So we have to have some constraint. So mathematically, we can cut through say a plane through this whole coding space. And I'll put this, I thought it's easier, but I figured out it's not really easy to understand in this way, right? But anyway, you have a way you can pick what we call the vector coding. By all these vectors to be differentiable, then it's becoming a legitimate code. I just show some pictures. So you can code 30 plex easily with one single shot, 4-channel single shot. And that's the whole brain you can actually decode it into 30 genes. You can do 30 genes for mouse embryos because it's so rapid. This whole process takes only a few hours, so you can do more if you want. So you can easily increase this total count to over like 100 million. Also, the cell number can be a few millions as well. And if you compare with other methods, this method gives you much cleaner background. So this is good. So you don't get confused like what's the background look like, why show some signal on the background. And you can do cancer. It's much. It's very noticeable. It's background not free. It's give you very high background. So hard to work, but it still work pretty well. You can differentiate different cell types. You can do this like Gaston to see the special features and pick the ROIs and do the further analysis. You can also take the continuous sectioning, like 20 sections, to do this like a quasi three-dimensional investigation. You can see how these different cell modules are wrinkled with each other. You can also see the B cells that form this network. But if you do only two-dimensional, it looks like separated columns from each other, but it's not. It's all connected together. You can do true three-dimensional using confocal or two-photon microscope. You can easily just penetrate 100 microns and do true three-dimensional. You can segment it into different cells, and you can also expand it to 64 plaques by a single shot easily. So it's a mouse brain. It's also a mouse slice, brain slice, 140 microns thick. Use the light sheet microscope, go through it. A few hours, you get a whole image, three-dimensional. So I just show these pictures, and here, thank you very much. I think I'm good, right?
Thank you very much, and big applause to all the speakers in this session. And now it's my pleasure to hand over to the next session, who is where we celebrate our partnership between the Human Cell Atlas and the UNESCO.
Hello, everyone. I'm John Randall. I'm the Executive Director of HCA, and I'm really excited to be introducing the speaker for the last presentation that we have before lunch. Lunch is indeed right around the corner, I promise you, both figuratively and physically. But before we get there, we have one more presentation, and then I'm actually going to ask all of you to remain seated because we are then going to follow up the presentation with a group photo, and we'll get instructions from the photographer about where he would like us all to be standing for that photo. Like Sarah mentioned earlier this morning, I was really honored to be part of an HCA delegation that traveled to Paris earlier this year to attend a UNESCO conference and ministerial level dialogue on science diplomacy. And while there, we signed a letter of intent with UNESCO to explore a major new partnership to support many of the areas of mutual interest and our shared principles and values. And as you'll hear from our next speaker, those overlaps and that alignment is really quite extensive. And so we're very excited, all of us at HCA, to be exploring how we could work together to advance our mutual objectives. And I'm very honored to be introducing Dr. Casimiro Vizzini from UNESCO, who will be explaining how many of UNESCO's programs do fit into what we are doing here in HCA. Like many of you, many people in HCA, he has a very interesting and impressive interdisciplinary background. He is a physician and also an expert in international diplomacy and capacity building, and really an ideal person for us to be working with, as well as his wonderful colleagues at UNESCO. So, Casimiro, please.
Thank you very much, and thank you, John and his team, for inviting us and myself. It's a great pleasure and an honor to represent UNESCO here with you. So this partnership, it's not symbolic, it's strategic. We are trying to foster the role of the Human Cell Atlas within the mandate of UNESCO as a true global public good. This is a quote from our Assistant Director General for Science, for Natural Sciences, Lydia Brito, and it's about science diplomacy. Science diplomacy is not a side product. It's how we build bridges in fragmented times, especially in this geopolitical turmoil, by making science a space for trust, cooperation, and shared progress. That's the spirit of this partnership. UNESCO in a role in global science cooperation. It's often strange for people to hear that UNESCO is working in science, and because it's often forgotten that the S of UNESCO stands for science. And because, I mean, the cultural heritage is much more known as a UNESCO mandate, but natural science has been since the inception of the organization in the 40s one of the main soul of the organization, and UNESCO acts as both standard setter and a platform for multilateral collaboration. I think we should keep our trust in multilateralism, especially nowadays. UNESCO, as an example in science, has a long-standing work in genetics and genomics. UNESCO led the 1997 Universal Declaration on the Human Genome and Human Rights. We hosted and supported the Human Variant Project. I was actually the coordinator of this project a few years ago, offering a neutral platform to ensure that countries from all regions could shape and benefit from data sharing standards. And this legacy shapes our work with the Human Cell Atlas. The cooperation, our partnership is essential because the Human Cell Atlas is transforming biomedical sciences, and UNESCO involvement reinforces this potential to deliver equitable benefits by enabling the project in global cooperation, inclusive participation, and fair access to data and knowledge. The partnership was formalized, as John said, during the 2025 UNESCO Science Diplomacy Conference. That was an opportunity to connect the Human Cell Atlas with shared global priorities for science and development. How do we frame the UNESCO contribution? Is through three main frameworks. Science diplomacy, as I said, this is how we use science to promote peace and cooperation. Open science, our global standard for equity, transparency, and access, and the UN Decade for Science for Sustainable Development that represent the horizon for turning knowledge into action for humanity and the planet. Talking about open science, UNESCO 2021 recommendation Open Science defines open science as the making scientific knowledge openly available, accessible, and reusable for everyone, opening the creation and communication of science to all of society. This means data must be open, but also inclusive and interoperable. Research must be collaborative, crossing borders and disciplines. Science must serve the collective good, not just individual discovery, and this is the approach we're bringing to the Human Cell Atlas. Science diplomacy, as I said, UNESCO has a very long story of science diplomacy. You can see a picture of the conference in March, and some milestones in science diplomacy were CERN, and that was one of the earliest initiative on science diplomacy after the Second World War, and Sesame, that is the synchrotron light facility in Jordan. This is also important in a region in which many of countries, I mean, some countries don't have diplomatic relations, and find in science the neutral space to collaborate. And then, as I mentioned before, the Human Variant Project, because I like to say that CERN and Sesame were large infrastructure investment, science cooperation, science diplomacy tools, but sharing data is something that nowadays is even more important, because it's not just about the infrastructure, but the culture of sharing. And with the Human Cell Atlas, we continue in this tradition, helping to ensure a large-scale biomedical science to become a platform for inclusion and not just discovery. We don't just promote science diplomacy, we practice it at UNESCO. And then finally, the decade of science for sustainable development, 2024-2033, calls for transformative action. It's about enabling science in every effort for sustainability with equity, participation, and transparency. Through the partnership with the Human Cell Atlas, the Human Cell Atlas became a flagship of that vision, connecting biology, health, and inclusive governance with the SDG agenda. I will try to be shorter because I'm getting too long. This is where we are advancing together. So first, alignment with UNESCO Open Science Principles. It's a mutual benefit. So we will use the Human Cell Atlas to enrich the toolkits on open science. Second, we have capacity development. We aim to ensure the researchers everywhere, particularly in underrepresented region, to contribute to the Human Cell Atlas, and including training, institutional partnership, and knowledge exchange. And finally, political visibility and international advocacy. Through UNESCO Intergovernmental Platform, we are helping to bring the Human Cell Atlas to the attention of ministers, regional bodies, and global science policy spaces. So this is about UNESCO network that can be used for our goal, and UNESCO has a category 2 category 2 centers and regional hubs that are extremely important to the work of UNESCO. And this is how we build capacity through training, policy engagement, technical infrastructure, of course, focusing on underdeserved region, not just as beneficiaries, but also as full actors in the Human Cell Atlas. And finally, we are, it's on the heart of UNESCO environmental sciences, so, trying to link the Human Cell Atlas with other international initiatives, and especially linked to environmental sciences. We are working to help these connections and find synergies with biodiversity initiatives, supporting planetary health agendas, and framing cellular data in global sustainability debates. And UNESCO unique mandate on this enables this kind of connections. This is what I think about what I said in general, sort of summary, and it resumes our will and our strong engagement with the Human Cell Atlas from UNESCO. And I finally will let you go for lunch. Thank you very much, and it was a great honor.
Okay, so we'll now have a group photo and then we'll go to lunch. I want to give you a couple instructions for lunch while we're getting set up. Lunch is outside in the exhibit space, so we encourage you to just get lunch. You can circulate. We are, you can use the breakout rooms if you need a place to sit for lunch. We just ask that you remove all your trash from the breakout rooms before you're done. And the first afternoon session will be a really exciting keynote from Shoma Richardri, and we ask you to be back here at 1:55, not 2 o'clock, 1:55, so that we can get started on time. Oh, yes, and I'm really pleased that there's been so much traffic already with the posters, but also we're doing the posters as well. They're open for viewing during the lunch period as well. So thank you all, and where's the photographer? Ah, yes, there you go.